Welcome to the Optimal Life Project After Dark series. Right now, as you listen to these words, millions of processes are unfolding inside you. Chemical messengers crossing synapses, neurotransmitters rising and falling like tides, hormones flooding through capillaries, carrying instructions to cells you'll never consciously know exist. And somewhere in all of this magnificent biological orchestration, your body is keeping score of something remarkably simple, yet profoundly powerful. How much sleep you've had. Tonight, we're exploring what happens when that score tips into deficit. Not chronic sleep deprivation measured in weeks or months, but something far more universal and immediate. What happens after just one bad night? One restless evening of tossing and turning. One night when stress kept your mind racing until 3 a.m. One 24-hour period where sleep simply didn't come. The research on this single night's impact is both humbling and fascinating. Within hours of inadequate sleep, measurable changes cascade through nearly every system in your body. Your brain chemistry shifts, your metabolism stumbles, your immune cells alter their behavior, your emotional regulation centers lose their equilibrium, and perhaps most surprisingly, many of these changes happen without you even realizing they're occurring. Over the next hour and a half, we'll journey through the immediate consequences of acute sleep deprivation, examining what current research reveals about this common yet underestimated challenge. We'll explore the cognitive fog that settles over your thinking, the metabolic chaos that disrupts your blood sugar, the emotional volatility that heightens your reactions, and the immune changes that leave you more vulnerable. We'll look at how your heart responds, how your physical coordination suffers, and how your brain's fundamental chemistry changes in response to staying awake. But this isn't a story of doom. This is a story of recognition and recovery. Because understanding what's happening in your body after one bad night doesn't just satisfy curiosity, it gives you power. The power to recognize when you're impaired, the power to make adjustments, the power to prioritize recovery, and the power to know that yes, your body can bounce back. As you settle in, know that these aren't abstract concepts. This is your biology, your chemistry, your lived experience translated into the language of neuroscience and metabolism. Every effect we discuss is something your body has likely experienced, perhaps without you fully understanding what was happening beneath the surface. Let's begin. The immediate cascade, understanding what starts happening. The moment you wake up after insufficient sleep, your body already knows something is wrong. It may take your conscious mind several minutes, perhaps even hours, to fully register the deficit. But on a cellular and systemic level, the changes have already begun. Sleep isn't merely the absence of waking. It's an active biological state during which your body performs essential maintenance, restoration, and preparation for the day ahead. During normal sleep, your brain cycles through distinct stages roughly every 90 to 120 minutes. Each stage serves specific purposes. The deeper stages of non-rapid eye movement sleep see your brain glucose utilization drop by 30 to 40% compared to waking levels, allowing neural tissues to rest and restore. During these deep sleep periods, growth hormone surges, supporting tissue repair. Your body temperature drops slightly, your blood pressure decreases by 10 to 20% in a phenomenon called nocturnal dipping, and your heart rate slows. The parasympathetic nervous system, often called the rest and digest system, becomes more active while the sympathetic fight-or-flight response quiets. When you miss out on adequate sleep, all of these restorative processes are truncated or eliminated. The consequences begin immediately, though they unfold in a predictable sequence. Research using multiple measurement techniques, including brain imaging, hormone assays, performance testing, and continuous physiological monitoring paints a detailed picture of how the body responds to acute sleep loss. Within the first few hours after inadequate sleep, several key changes are already measurable. Adenosine, a neuromodulator that accumulates in your brain during waking hours, reaches higher concentrations than it would after proper sleep. Normally, adenosine levels build throughout the day, creating what researchers call sleep pressure, the biological drive to sleep. During sleep, adenosine levels decrease, resetting this pressure. Research suggests that when sleep is insufficient, adenosine continues accumulating or fails to clear adequately, which could leave your brain operating under persistent inhibitory pressure. This adenosine accumulation appears to have widespread effects, particularly in brain regions like the basal forebrain and prefrontal cortex. Studies indicate that elevated adenosine inhibits wake-promoting neurons and reduces the release of acetylcholine, a neurotransmitter crucial for attention and learning. Based on these findings, it seems your brain operates in a state somewhere between fully awake and genuinely asleep, a twilight zone of compromised function. Concurrently, your hormonal landscape begins shifting. Cortisol, the stress hormone that typically peaks in early morning to help you wake up, may show altered patterns after poor sleep. Some studies report elevated evening cortisol levels following sleep deprivation, while the normal morning peak might be blunted. This disruption in cortisol rhythm appears to affect everything from inflammation to blood sugar regulation to immune function. Your sympathetic nervous system, responsible for arousal and the fight-or-flight response, also shows changes. Some research suggests increased sympathetic activity after sleep loss, though findings vary depending on the severity of deprivation and individual differences. What seems consistent is that the delicate balance between sympathetic activation and parasympathetic calming becomes disrupted, leaving you physiologically on edge even when you're trying to relax. These early changes set the stage for everything that follows. They create a cascade effect where one disrupted system influences another, which affects a third, until nearly every aspect of your physiology shows some degree of alteration. The remarkable part is how quickly this happens. We're not talking about weeks of poor sleep. We're talking about hours. Research participants undergoing controlled sleep deprivation studies, where scientists can precisely measure changes before and after a single night of reduced or absent sleep, consistently show that cognitive performance begins declining within hours. Reaction times slow. Attention becomes less focused. The ability to sustain concentration wavers. And critically, people often don't realize the extent of their impairment. This disconnect between objective performance and subjective perception is one of the most dangerous aspects of sleep deprivation. When researchers ask sleep-deprived individuals to rate their own sleepiness and performance, the ratings often fail to match objective test results. Someone might report feeling only moderately tired, while their reaction time has slowed by hundreds of milliseconds and their error rate has doubled. This perception gap means you might not recognize when you're impaired, leading to decisions and behaviors that increase risk. The brain regions most affected by acute sleep loss include the prefrontal cortex, which handles executive functions like planning, decision-making, and impulse control. Imaging studies using functional MRI show decreased activation in the dorsolateral prefrontal cortex and the intraparietal sulcus during attention tasks after sleep deprivation. These regions light up less brightly on brain scans, indicating reduced activity when sleep-deprived people attempt cognitively demanding tasks. Simultaneously, other brain regions may show increased or compensatory activation. The temporal lobes might reduce their activity, while parietal regions increase theirs, suggesting the brain attempts to compensate for prefrontal deficits by recruiting alternative neural pathways. This compensation is rarely sufficient to maintain normal performance, but it does indicate your brain's remarkable flexibility in trying to cope with adverse conditions. Another early change involves your emotional regulation centers. The amygdala, a small almond-shaped structure deep in your brain responsible for processing emotions, particularly negative ones, begins showing altered activity patterns. Research using brain imaging has found that after just one night of total sleep deprivation, the amygdala shows approximately 60% greater activation in response to negative emotional stimuli compared to the rested state. This finding has been replicated across multiple studies, suggesting a robust effect. This amygdala hyperreactivity wouldn't necessarily be catastrophic if your prefrontal control systems remained fully functional. After all, feeling emotions strongly isn't inherently problematic if you can regulate them effectively. The difficulty arises because the same sleep deprivation that amplifies amygdala responses also weakens the prefrontal cortex's regulatory influence. Studies show that the functional connectivity between the medial prefrontal cortex and the amygdala, the neural pathway that normally allows top-down emotional control, becomes disrupted after sleep loss. It's as if the accelerator on your emotions is pressed down while the brakes aren't working properly. This emotional vulnerability manifests in ways you've probably experienced. Irritability increases. Minor frustrations feel disproportionately overwhelming. A colleague's neutral comment might be interpreted as criticism. A small inconvenience triggers disproportionate frustration. You might find yourself tearing up at advertisements or feeling unreasonably angry at trivial annoyances. These aren't character flaws or signs of weakness. They're predictable neurobiological consequences of acute sleep loss. The same neural changes that affect emotion also influence how you perceive and respond to rewards. Research indicates that sleep deprivation amplifies reactivity in the brain's reward networks, particularly in regions like the striatum and ventral tegmental area. This heightened reward sensitivity might sound positive, but studies suggest it actually leads to impulsive decision-making and poor risk assessment. You become more drawn to immediate gratification and less capable of weighing long-term consequences. Studies have shown that sleep-deprived individuals are more likely to choose risky options in gambling tasks, more likely to purchase unnecessary items, and more likely to consume high-calorie foods. Drawing from these observations, researchers propose that this involves dopamine receptor changes and altered functional connectivity between reward centers and decision-making regions. Your brain's reward system goes into overdrive while the rational evaluator sits in the back seat, occasionally offering weak objections that the dopamine-flooded reward system easily ignores. These immediate changes, unfolding within hours of inadequate sleep, form the foundation for all the specific effects we'll explore in depth. They represent your body's attempt to function under suboptimal conditions to maintain homeostasis when a critical input, sleep, has been denied. Understanding this cascade helps explain why a single bad night can affect so many seemingly unrelated aspects of your functioning. It's all connected through these fundamental shifts in brain chemistry, hormonal balance, and neural activation patterns. As we move forward, we'll examine each affected system more closely, understanding not just that changes occur, but how they manifest, what mechanisms drive them, and what it all means for your daily life. We'll start where you probably notice the effects first, and most obviously, in your ability to think clearly and respond quickly. We'll explore the fog that descends over your cognition when sleep is insufficient. The foggy mind, cognitive impairment after one night. You wake up after 4 hours of sleep, or perhaps after a restless night of frequent awakening. You lie in bed for a moment, taking stock. You feel tired, certainly, but you tell yourself you've functioned on less. You shower, dress, eat breakfast, and head into your day. Maybe you feel a bit sluggish, but overall, you think you're managing. Then you sit down to work. You read the same paragraph 3 times without absorbing it. You reach for a word that usually comes easily, but it hovers just out of reach. You meant to respond to an email, but found yourself staring at social media instead. Simple tasks feel inexplicably difficult. Complex problems seem overwhelming. And throughout it all, you might not fully recognize how impaired you actually are. This is the cognitive reality of acute sleep deprivation. The effects are measurable, substantial, and remarkably consistent across individuals, even though the subjective experience varies. Let's start with attention, arguably the cognitive function most obviously and immediately affected by sleep loss. Attention isn't a single thing, it's a collection of related processes. Sustained attention involves maintaining focus on a task over time. Selective attention means filtering out distractions to concentrate on what matters. Divided attention is your ability to track multiple things simultaneously. After poor sleep, all of these attentional capacities decline, though not equally. Research using the psychomotor vigilance test, a simple reaction time task that requires maintaining attention and responding to randomly appearing stimuli, consistently shows degraded performance after sleep deprivation. Reaction times slow, sometimes by hundreds of milliseconds. But more importantly, attentional lapses increase dramatically. These lapses, moments when you simply don't respond to a stimulus you should have noticed, can increase from nearly 0 in rested individuals to dozens per testing session in sleep-deprived ones. These lapses aren't merely slowness. They're complete failures of sustained attention. Micro-episodes where your brain briefly disengages from the task. During such a lapse, a stimulus could appear right in front of you, and you wouldn't register it. In laboratory settings, this means missing a test signal. In real life, this could mean not seeing the brake lights of the car ahead of you or missing a critical piece of information in a conversation. Studies examining 1 night of sleep restriction, typically defined as 4 to 6 hours of sleep, or 1 night of total sleep deprivation, show a consistent pattern. Sustained attention, the kind required for monotonous tasks like driving long distances or monitoring equipment, deteriorates measurably. The degree of impairment depends on sleep duration during the restriction period. Getting 4 hours seems better than getting 2, but neither approaches normal rested performance. Interestingly, simple reaction time tasks, those requiring just a single quick response, sometimes show smaller effects than more complex attention tasks. This suggests that while your basic processing speed might be preserved relatively well, at least in the short term, your ability to maintain that speed consistently over time or across more demanding situations suffers more dramatically. Selective attention, your ability to focus on relevant information while ignoring distractions, also shows impairment. Studies using Stroop tests, where participants must name the color of ink used to print words while ignoring the actual word, show increased interference effects after sleep deprivation. It takes longer to correctly identify colors when the word itself names a different color, suggesting that your attentional filter isn't working as effectively. Distractions break through more easily, pulling your focus away from what you intended to concentrate on. Working memory, the mental workspace where you temporarily hold and manipulate information, represents another casualty of poor sleep. Working memory is what allows you to remember a phone number long enough to dial it, to follow the thread of a conversation while formulating your response, or to solve a multi-step problem without losing track of where you are. Research findings on working memory effects vary somewhat depending on the specific test used and the severity of sleep deprivation, but several patterns emerge. Tasks requiring the maintenance and manipulation of information, particularly complex ones like the N-back task where you must remember items from N steps back in a sequence, show clear impairment after sleep loss. The more demanding the working memory load, the greater the performance decline. Brain imaging during working memory tasks reveals reduced activation in the dorsolateral prefrontal cortex after sleep deprivation, the same region that shows reduced activity during attention tasks. This makes biological sense. Working memory and attention are deeply intertwined processes that rely heavily on prefrontal cortex function. When that region is operating under the inhibitory pressure of accumulated adenosine and hormonal disruption, both attention and working memory suffer. Episodic memory, your ability to encode and later recall specific experiences and information, faces particular vulnerability during sleep deprivation, though the effects manifest differently depending on when the deprivation occurs. Sleep after learning appears crucial for consolidating new memories, transforming them from fragile temporary traces into more stable long-term representations. When you learn something new, initial encoding involves the hippocampus, a seahorse-shaped structure deep in your temporal lobe. During subsequent sleep, particularly during deep non-REM sleep and REM sleep, neural activity patterns from the learning episode reactivate, and connections between the hippocampus and neocortex strengthen. This process, called consolidation, integrates new information into your existing knowledge networks. If you're sleep-deprived before learning, research suggests your ability to initially encode information may be compromised. The hippocampus doesn't function as effectively, and the attentional deficits we've discussed mean you don't process the to-be-learned material as deeply. If you're sleep-deprived after learning, consolidation appears to suffer. The neural replay and synaptic strengthening that should occur during sleep are truncated or absent. Studies examining memory for word lists, story narratives, or spatial locations consistently show that sleep-deprived individuals remember less than their rested counterparts. The effect size varies. Some studies show modest impairment while others demonstrate substantial deficits, but the direction is consistent. Less sleep equals worse memory. One particularly concerning finding involves false memories. Research indicates that sleep deprivation increases the likelihood of forming inaccurate memories, recalling events that didn't actually happen or details that weren't actually present. This appears related to impaired encoding quality and reduced ability to discriminate between actual experiences and similar but distinct alternatives. Your memory system, operating under duress, fills in gaps with plausible but incorrect information. Decision-making and executive function, the higher-order cognitive processes that allow you to plan, problem-solve, and control your behavior, show perhaps the most complex pattern of sleep deprivation effects. Some aspects of decision-making hold up surprisingly well, while others collapse dramatically. Routine decisions following established rules often remain relatively intact after moderate sleep loss. If you're performing a familiar task with clear guidelines, you can often execute it adequately, even when sleep-deprived, particularly if the task doesn't require sustained attention over long periods. Your procedural knowledge, the know-how embedded in practiced routines, appears somewhat protected. However, decisions requiring flexibility, creativity, or dealing with novel situations show marked impairment. Sleep deprivation seems to make people more rigid in their thinking, more likely to perseverate on previous strategies even when those strategies are no longer effective. The ability to generate alternative solutions, to think divergently, and to adapt to changing circumstances deteriorates. Risk assessment also becomes skewed. Multiple studies demonstrate that sleep-deprived individuals underestimate risks and overvalue potential rewards. In gambling tasks, Sleep-deprived participants make riskier bets. In economic decisions, they show reduced loss aversion, meaning they're more willing to accept potential losses in pursuit of gains. This isn't confidence, it's impaired judgment stemming from the altered functional connectivity between prefrontal control regions and reward centers we discussed earlier. Moral judgment, the kind of ethical reasoning required for complex social and professional decisions, appears to take longer and become more difficult after sleep deprivation. Studies examining responses to moral dilemmas show increased response times, suggesting sleep-deprived individuals struggle more to integrate cognitive and emotional information when making ethical decisions. The quality of moral reasoning may not necessarily decline, but the cognitive effort required increases substantially. Language processing also shows interesting effects. Verbal fluency, your ability to quickly generate words meeting specific criteria, tends to show small to moderate impairment after sleep loss. It takes longer to think of words starting with a particular letter or belonging to a specific category. However, more automatic language processes, like understanding common words or using grammar correctly in spontaneous speech, remain relatively preserved unless sleep deprivation is extreme. Attention to these language effects becomes important in professional contexts. A presentation you give after poor sleep might contain more filler words, longer pauses as you search for vocabulary, and less fluid transitions between ideas. Your writing may become less eloquent, with simpler sentence structures and more repetition. These aren't catastrophic failures, but they represent subtle degradations that can impact how effectively you communicate. Perception itself can be affected by sleep deprivation, though usually only with severe or prolonged sleep loss. After one night of moderate sleep restriction, you're unlikely to experience full hallucinations or major perceptual distortions. However, more subtle changes can occur. Visual perception may become less sharp. You might have more difficulty distinguishing fine details or reading small print. Color discrimination could be slightly impaired. The overall quality and speed of perceptual processing decline. Perhaps most concerning is the phenomenon of microsleeps, brief episodes lasting from a fraction of a second to several seconds where you essentially fall asleep momentarily, even while appearing to be awake. During a microsleep, your brain waves briefly show sleep-like patterns. Your eyelids may droop or close completely, and your responsiveness to external stimuli disappears. Microsleeps are particularly dangerous because they're often preceded and followed by periods of normal waking, creating the illusion that you're fully alert. You might experience a microsleep while driving, completely missing several seconds of road time, yet have no memory of having lost consciousness. This explains why drowsy driving is so treacherous. You genuinely might not realize you're impaired until it's too late. These microsleeps appear related to localized sleep intrusion into wake. Research suggests that while parts of your brain remain awake, other regions, particularly those under the heaviest adenosine pressure, may briefly enter sleep states. Your brain becomes a patchwork of sleeping and waking regions, and during moments when critical networks enter sleep, cognitive function and behavioral responsiveness cease. The cumulative effect of all these cognitive impairments is substantial. After just one night of inadequate sleep, you're operating with measurably reduced attention, slower reaction times, compromised working memory, impaired learning ability, degraded decision-making quality, and increased vulnerability to microsleeps and attentional lapses. You might compensate through increased effort, and you might not notice all of these deficits subjectively, but they're present and measurable. Studies directly comparing performance after one night of total sleep deprivation to performance while legally intoxicated by alcohol have found similar levels of impairment on certain tasks. Research indicates that being awake for 24 hours produces cognitive and motor impairments roughly equivalent to a blood alcohol content of 0.05 to 0.08% on some measures. This isn't hyperbole or exaggeration, it's what controlled laboratory studies demonstrate, though the specific tasks and exact equivalents vary across studies. Yet we routinely operate after poor sleep in contexts where we'd never operate while intoxicated. We drive, we care for children, we perform surgery, we make critical business decisions, all while cognitively impaired to a degree we'd consider completely unacceptable if caused by anything other than sleep deprivation. Understanding the extent and nature of cognitive impairment after poor sleep serves two purposes. First, it validates your experience. If you feel foggy and ineffective after a bad night, that's not weakness or imagination. It's neurobiology. Second, it emphasizes the importance of recognizing impairment and making appropriate adjustments, recognizing when tasks should be postponed, when extra care is needed, and when pushing through isn't heroic, but dangerous. As we transition to our next section, keep in mind that cognitive function doesn't operate in isolation. The metabolic changes occurring simultaneously with these cognitive deficits not only affect your physical health, they feed back to influence cognition. The tired mind and the metabolically disrupted body are part of a unified system, each affecting the other in a complex web of interactions. Metabolic chaos. Blood sugar, insulin, and appetite gone haywire. While your brain struggles with cognitive fog and emotional volatility, your metabolic system, the intricate network of processes that converts food into energy and regulates your internal chemistry, enters its own state of disruption. What's remarkable about the metabolic effects of sleep deprivation is how quickly they manifest and how closely they mirror the early stages of serious metabolic disease. In essence, one night of poor sleep can temporarily create a pre-diabetic-like metabolic state in healthy individuals. Let's begin with glucose metabolism, the process by which your body regulates blood sugar levels. Under normal circumstances, when you eat, blood glucose rises. Your pancreas responds by releasing insulin, a hormone that signals cells throughout your body to take up glucose from the bloodstream. This system normally operates with remarkable precision, keeping blood sugar within a relatively narrow range despite dramatic variations in food intake and energy expenditure. After inadequate sleep, this precision falters. Multiple research studies using sophisticated measurement techniques like hyperinsulinemic euglycemic clamps and continuous glucose monitoring have documented that even a single night of partial sleep restriction, sometimes as little as 4 hours of sleep, impairs glucose metabolism in ways that can be measured the very next day. One study examining healthy young adults found that after one night of 4 hours of sleep, glucose tolerance decreased significantly. When given a standard oral glucose tolerance test the following day, glucose levels remained elevated longer than after normal sleep. From these findings, researchers infer that cells aren't taking up glucose as efficiently, leaving more sugar circulating in the blood for extended periods. The underlying mechanism appears to involve insulin resistance, a condition where cells become less responsive to insulin's signal. After sleep restriction, cells in your liver, muscles, and fat tissue don't respond as robustly to insulin. Consequently, your pancreas must produce more insulin to achieve the same glucose-lowering effect. Studies using clamp techniques, where researchers maintain constant blood glucose while measuring how much insulin is required, show that insulin sensitivity can decrease by approximately 20 to 30% after just one night of restricted sleep, though individual responses vary. This insulin resistance manifests in several ways. Hepatic glucose production, the liver's creation and release of glucose into the bloodstream increases during sleep deprivation and remains elevated the following day. Normally, insulin suppresses hepatic glucose production. When insulin resistance develops, this suppression becomes less effective, and the liver continues releasing glucose even when blood sugar is already adequate or elevated. Peripheral insulin sensitivity, the responsiveness of muscle and fat tissue to insulin, also decreases. Studies show reduced glucose disposal rates during hyperinsulinemic clamps after sleep restriction. This means that even when insulin levels are high, muscles aren't taking up glucose as effectively as they should. The result is that glucose lingers in the bloodstream, and more insulin is required to eventually clear it. Importantly, some research indicates these metabolic changes can occur even after partial sleep deprivation, not just total sleep loss. Getting 5 or 6 hours instead of 8 may be sufficient to trigger measurable alterations in glucose homeostasis. The severity of impairment generally correlates with the degree of sleep loss, but even moderate restriction appears sufficient to induce detectable changes. The timing of sleep also matters. Studies examining delayed sleep onset versus early morning awakening suggest different effects on metabolism. Late-night partial sleep deprivation, where you stay up late and get only a few hours of sleep in the early morning, appears to produce more pronounced metabolic disturbances than early evening sleep restriction followed by early morning waking. This may relate to disruption of circadian rhythms, the 24-hour internal clock that regulates numerous metabolic processes. Beyond glucose and insulin, sleep deprivation dramatically affects appetite-regulating hormones, creating conditions that promote increased food intake. Two key hormones, leptin and ghrelin, become dysregulated in ways research suggests drive eating behavior. Leptin, produced by fat cells, acts as a satiety signal. When leptin levels are high, you feel full and satisfied, and your brain reduces the drive to eat. When leptin is low, hunger increases. Studies consistently show that sleep restriction decreases leptin levels. After even a few nights of inadequate sleep, leptin production drops, which could remove the biological brake on appetite. Ghrelin, produced primarily in the stomach, works in the opposite direction. It's an appetite-stimulating hormone, often called the hunger hormone. Ghrelin levels rise before meals, promoting eating, and drop after eating. Research demonstrates that sleep deprivation increases ghrelin levels, amplifying hunger signals. The combination of decreased leptin and increased ghrelin creates a powerful biological drive to eat. Studies examining these hormonal changes alongside actual food intake show that sleep-deprived individuals don't just feel hungrier, they actually consume more calories, often several hundred extra calories per day. The increase isn't random either. Research indicates that sleep-deprived people show particular cravings for high-carbohydrate, high-fat foods, the exact types that contribute most to weight gain and metabolic dysfunction. Brain imaging studies help explain these cravings. When researchers scan the brains of sleep-deprived individuals while showing them images of various foods, reward centers show enhanced activation in response to high-calorie options. The sight of pizza, cookies, or ice cream triggers stronger reward responses than it would in rested individuals. Simultaneously, decision-regulating regions in the prefrontal cortex show reduced activity, which suggests the rational evaluation of whether eating that food aligns with your health goals is compromised. This isn't willpower failure, it's neurobiology. Your brain's reward system is amplified, your prefrontal control is weakened, and your appetite hormones are signaling for you to eat calorie-dense foods. Resisting these biological pressures requires extraordinary effort, and most people, quite reasonably, don't succeed in resisting them completely. Some research suggests that endocannabinoid signaling, the same system affected by cannabis, may be involved in sleep deprivation-induced food cravings. Sleep loss appears to increase endocannabinoid levels, which could enhance the pleasurability of eating and specifically increase desire for palatable, high-calorie foods. This might explain why sleep-deprived eating feels particularly gratifying, reinforcing the behavior. Energy expenditure, the number of calories your body burns, doesn't appear to increase enough to compensate for the increased food intake. Some studies find modest increases in energy expenditure during sleep deprivation, perhaps reflecting the metabolic cost of staying awake longer. However, this increase is typically much smaller than the increase in calorie consumption. The net effect is positive energy balance, meaning you're consuming more calories than you're burning, promoting fat storage and weight gain. Physical activity often decreases during and after sleep deprivation, further contributing to energy imbalance. When you're tired, you're less likely to exercise, less likely to engage in active leisure pursuits, and more likely to be sedentary. Every extra hour spent sitting on the couch instead of moving around represents additional missed energy expenditure. The metabolic consequences extend beyond glucose and appetite regulation. Lipid metabolism, the processing of fats in the bloodstream, also shows alterations after sleep deprivation. Some studies report increased free fatty acid levels, changes in triglyceride concentrations, and alterations in cholesterol metabolism. While a single night might not produce dramatic lipid changes, the direction of these changes, when they occur, generally points toward increased cardiovascular risk. Cortisol, which we mentioned earlier in the context of stress response and circadian disruption, also influences metabolism. Elevated cortisol, particularly in the evening when it should be low, promotes insulin resistance, increases blood sugar, and can drive visceral fat accumulation. The cortisol alterations observed after sleep deprivation thus contribute directly to the metabolic dysregulation we've been discussing. Growth hormone secretion, which normally surges during deep sleep, may be affected by sleep disruption. Extended periods with elevated growth hormone can paradoxically impair glucose metabolism by reducing insulin sensitivity. If sleep disruption alters the typical pattern of growth hormone secretion, extending its presence into times when it normally would be low, this could contribute to the observed insulin resistance. Thyroid function, another key regulator of metabolism, can also be affected by sleep deprivation, though effects tend to be more pronounced with chronic rather than acute sleep loss. Thyroid hormones influence metabolic rate, and even modest alterations could affect energy balance. One night of poor sleep likely isn't sufficient to produce major thyroid dysfunction, but it represents one more system showing subtle dysregulation. Inflammatory markers, chemical signals of inflammation in the body, increase after sleep deprivation. C-reactive protein, interleukin-6, and tumor necrosis factor alpha, all markers associated with systemic inflammation, show elevations following sleep loss. These inflammatory changes don't just reflect immune system activation, they actively impair metabolic function. Inflammation in metabolic tissues like liver, muscle, and fat disrupts insulin signaling and promotes insulin resistance. The cumulative picture is of a metabolic system thrown into disarray. Glucose stays elevated longer. More insulin is required to clear it. Appetite increases dramatically. Food cravings intensify, particularly for foods that worsen metabolic health. Energy expenditure doesn't rise to match increased intake. Physical activity declines. Inflammatory signals rise. Multiple hormonal systems shift away from their normal patterns. What makes this especially concerning is the similarity between these acute effects and the chronic metabolic dysfunction seen in prediabetes and type 2 diabetes. The glucose tolerance impairment observed after one night of 4 to 5 hours of sleep can resemble that of people with prediabetes. The insulin resistance can approach levels seen in early metabolic disease. Of course, These effects are typically reversible with recovery sleep, while chronic metabolic diseases develop over years of sustained insults. But the fact that one night can temporarily produce disease-like metabolic states underscores how fundamental sleep is to metabolic health. For individuals already at metabolic risk, those with family histories of diabetes, those carrying excess weight, or those in the early stages of metabolic syndrome, the additional metabolic stress of poor sleep may be particularly consequential. It represents yet another factor pushing their already challenged metabolic systems further toward dysfunction. Some research suggests that the metabolic effects of sleep deprivation might persist even after 1 or 2 nights of recovery sleep. While cognitive function often bounces back relatively quickly with adequate sleep, metabolic parameters may take longer to fully normalize. This suggests a kind of metabolic memory, where the disruption leaves a lingering footprint even after the immediate insult has resolved. Understanding these metabolic effects provides crucial context for the broader health consequences of inadequate sleep. The increased cardiovascular disease risk associated with chronic sleep restriction likely operates partially through these metabolic pathways. The connection between short sleep duration and obesity almost certainly involves the appetite and metabolic dysregulation we've described. The heightened diabetes risk in habitual short sleepers probably reflects cumulative exposure to these acute metabolic insults. But knowledge is power. Recognizing that metabolic chaos follows sleep deprivation allows you to make informed choices. On days after poor sleep, you might be extra vigilant about food choices, knowing that your biology is conspiring to make you overeat junk food. You might prioritize recovery sleep to restore metabolic function. You might avoid situations where metabolic demands are highest, like intense physical training or major dietary changes. And you might treat sleep not as an optional luxury, but as a critical component of metabolic health, as essential as diet and exercise. As we move forward to explore the emotional and immune consequences of sleep loss, remember that all these systems are interconnected. The metabolic disruption influences immune function. Immune activation affects metabolism. And both metabolic and immune changes feed back to influence the brain, affecting mood, cognition, and behavior. The body is a unified system, and sleep deprivation disrupts that unity at multiple levels simultaneously. Emotional turbulence, mood, stress, and losing your cool. You snap at your partner over something trivial. A minor work setback feels catastrophic. You well up with tears at a sentimental commercial. Or perhaps you just feel irritable and on edge, unable to pinpoint exactly why, but aware that your emotional reactions seem disproportionate to the situations triggering them. Welcome to the emotional landscape of sleep deprivation. The emotional effects of inadequate sleep are among the most immediately noticeable to both the sleep-deprived individual and those around them. Unlike some cognitive impairments that require careful testing to detect, emotional dysregulation often announces itself unmistakably. Yet the biological mechanisms underlying these emotional changes are only beginning to be fully understood, and the research reveals a fascinating story about how sleep loss fundamentally alters the brain's emotional processing systems. At the center of this story sits the amygdala, a small almond-shaped structure deep in the temporal lobe of your brain. The amygdala acts as your brain's emotional sentinel, rapidly detecting emotionally significant stimuli, particularly threats and other negative information. When you encounter something potentially dangerous, emotionally charged, or socially significant, your amygdala responds quickly, initiating emotional and physiological responses before your conscious mind even fully processes what's happening. Under normal circumstances, the amygdala's responses are modulated by connections with the prefrontal cortex, particularly the medial prefrontal cortex. The prefrontal cortex provides top-down regulatory control, a kind of supervisory system that can dampen or modulate amygdala reactivity based on context, goals, and rational evaluation. This prefrontal amygdala circuit allows you to feel emotions while maintaining appropriate control over how you express and respond to them. After sleep deprivation, this crucial regulatory system becomes dramatically disrupted. One of the most well-replicated findings in sleep neuroscience involves the amygdala's response to negative emotional stimuli after sleep loss. Using functional MRI brain imaging, researchers have demonstrated that following one night of total sleep deprivation, the amygdala shows approximately 60% greater activation in response to negative emotional pictures compared to the rested state. This amplified amygdala response isn't just a statistical artifact visible only in averaged brain scans. It corresponds to subjective reports of heightened emotional reactivity. Sleep-deprived individuals report feeling more disturbed by negative images, more anxious, and more emotionally vulnerable. The brain imaging and the subjective experience align, both pointing toward genuine hyperreactivity in emotional processing. But amygdala amplification represents only half the story. Simultaneously with this increased emotional reactivity, the functional connectivity between the medial prefrontal cortex and the amygdala, the neural connection that normally allows prefrontal control of emotional responses, becomes weakened. Brain imaging shows reduced correlation in activity between these regions after sleep deprivation, suggesting that the prefrontal cortex isn't effectively regulating the amygdala's responses. Interestingly, while connectivity with the prefrontal cortex decreases, the amygdala shows increased connectivity with other brain regions, particularly the locus coeruleus, a brainstem structure involved in arousal and the fight-or-flight response. This shift in connectivity patterns suggests that after sleep loss, the amygdala becomes more strongly coupled to arousal systems and less constrained by rational control systems. Your emotional brain effectively gets more access to your body's alarm system while losing access to your rational supervisor. The consequences of this altered amygdala-prefrontal relationship manifest in multiple ways. Emotional regulation, the ability to modulate your emotional responses through conscious strategies, becomes more difficult after sleep deprivation. Tasks requiring cognitive reappraisal, where you deliberately reinterpret a situation to change your emotional response to it, show impaired effectiveness when you're sleep-deprived. You might know rationally that a situation doesn't warrant such a strong emotional reaction, but you struggle to actually implement that rational perspective. Studies using emotion regulation tasks show that sleep-deprived individuals require more time and more effort to downregulate negative emotions. Their success rate at regulating emotions decreases, and the subjective experience of regulation feels more difficult. This isn't merely fatigue making everything harder. The specific neural circuits required for emotional control are operating under suboptimal conditions. Interestingly, the effects of sleep deprivation on emotional processing aren't limited to negative emotions. Research also demonstrates that sleep loss amplifies responsiveness to positive stimuli as well, though this has received less attention. Brain reward networks, including the ventral striatum and ventral tegmental area, show enhanced activation in response to pleasurable or rewarding stimuli after sleep deprivation. This heightened reward sensitivity creates an interesting dynamic. You might feel more intensely upset by negative events while simultaneously feeling more drawn to potential rewards and pleasures. This combination can lead to impulsive behavior, seeking immediate gratification to escape negative feelings while lacking the prefrontal control to properly evaluate whether those reward-seeking behaviors serve your long-term interests. Food cravings, which we discussed in the metabolic section, represent one manifestation of this reward amplification. But the phenomenon extends beyond eating. Sleep-deprived individuals show increased monetary risk-taking in gambling tasks, greater attraction to hedonic activities, and increased impulsivity across various domains. The reward system says yes more loudly, while the control system's no becomes weaker. Stress reactivity also increases after sleep deprivation. Both psychological and physiological stress responses become exaggerated when you're under-rested. Heart rate and blood pressure may show larger increases in response to stressful situations. Subjective reports of stress and anxiety intensify. Recovery from stress takes longer, with physiological markers of stress remaining elevated for extended periods after the stressor has passed. Some research suggests an interaction between sleep deprivation and acute stress, where a stressor occurring during sleep deprivation produces even larger effects than either factor alone would predict. However, other studies find more complex patterns, including instances where acute stress during sleep deprivation actually improves some aspects of performance, possibly through arousal enhancement. The relationship appears to depend on stress timing, intensity, and the specific outcomes measured. Mood disturbances represent another prominent feature of the emotional landscape after poor sleep. Irritability increases. Feelings of fatigue and lack of energy intensify. Some people report feeling more depressed or sad. Others describe feeling more anxious or on edge. The specific mood changes vary somewhat between individuals, but overall mood typically worsens across multiple dimensions after sleep loss. Intriguingly, there's a paradoxical relationship between sleep deprivation and depression. While chronic sleep problems predict increased depression risk, acute sleep deprivation can temporarily improve mood in some people who are already depressed. This antidepressant effect of sleep deprivation has been documented for decades and remains something of a mystery. Current theories suggest it may involve alterations in the brain's serotonin system, changes in REM sleep patterns, or shifts in prefrontal amygdala connectivity that differ between depressed and healthy individuals. For non-depressed people, however, acute sleep deprivation generally worsens mood rather than improving it. The tired brain struggles to maintain emotional equilibrium, leading to increased negative affect and emotional volatility. Social cognition, your ability to understand and navigate social interactions, also suffers under sleep deprivation. Reading facial expressions becomes more difficult. Sleep-deprived individuals are more likely to misinterpret neutral faces as angry or threatening. They show reduced accuracy in identifying subtle emotional expressions. This creates potential for social misunderstandings and conflicts, as you might perceive criticism or hostility where none was intended. Empathy and perspective-taking, the abilities to understand and share others' emotions and to see situations from their viewpoints, appear to decline after sleep loss. You become more focused on your own immediate experiences and less capable of accurately modeling others' mental states. This self-focus, combined with increased irritability and emotional reactivity, can strain relationships and impair collaborative work. Theory of mind tasks, where you must infer what someone else knows or believes, show impairment after sleep deprivation. Your ability to maintain a complex model of others' perspectives and intentions falters when your prefrontal cortex is operating suboptimally. This can make social interactions feel more confusing and more effortful, adding to the overall sense of being overwhelmed and out of sorts. Emotional memory, the way emotions affect memory formation and retrieval, shows interesting changes after sleep loss. Normal sleep, particularly REM sleep, appears to play a role in processing emotional experiences, helping to consolidate the factual content of emotional memories while reducing their emotional intensity. This process, sometimes called emotional depotentiation, may help you remember what happened during emotional events without remaining as emotionally aroused by those memories. When sleep is disrupted, this emotional processing appears impaired. Emotional memories may remain more emotionally charged. The ability to discriminate between emotional and neutral information becomes less precise. And as we noted in the cognitive section, the likelihood of forming false emotional memories increases, where you might remember events as more emotionally intense than they actually were, or remember emotional details that didn't occur. Individual differences in vulnerability to these emotional effects are substantial. Some people show dramatic mood changes and emotional dysregulation after poor sleep, while others remain relatively stable emotionally, even when cognitively impaired. Age appears to matter, with some research suggesting younger adults may be more vulnerable to emotional effects of sleep loss than older adults, though findings are mixed. Personality traits, baseline emotional regulation skills, and previous experience with sleep deprivation may all influence how severely emotional functioning is affected. Gender differences have also been noted in some studies, though again findings are inconsistent. Some research suggests women may show greater emotional reactivity to sleep deprivation, while other studies find no gender difference or even opposite patterns. The variability across studies likely reflects the complexity of the systems involved and the multiple factors that influence emotional responses. Understanding the neural mechanisms helps make sense of your subjective experience. When you feel irrationally angry at a minor inconvenience after poor sleep, you're not being childish or overreacting in a purely psychological sense. Your amygdala is literally responding more intensely to negative stimuli, while your prefrontal cortex's ability to modulate that response is impaired. When you find yourself making impulsive decisions driven by immediate desires, it's because your reward system is amplified while your rational evaluator is weakened. This biological understanding doesn't eliminate responsibility for behavior, but it does provide context. It helps you recognize that extra caution is warranted after poor sleep, that situations requiring emotional control or thoughtful decision-making might best be avoided or approached with extra care. It validates the difficulty of maintaining composure when your brain's emotional regulation systems are operating under adverse conditions. For relationships, this understanding can be invaluable. Knowing that you or your partner is more emotionally reactive and less able to regulate emotions after poor sleep can help de-escalate conflicts. Instead of interpreting emotional overreactions as character flaws or signs of relationship problems, you can recognize them as temporary biological states resulting from sleep deprivation. This doesn't mean accepting all behavior without consequence, but it does allow more compassion and more appropriate responses. As we transition to examining how sleep loss affects the immune system, keep in mind that these emotional changes don't exist in isolation. Stress responses and emotional states directly influence immune function through neural and hormonal pathways. The emotional turbulence of sleep deprivation thus becomes one more factor in the cascade of effects rippling through multiple body systems. The immune shift, how one night alters your body's defenses. While you're navigating emotional turbulence and metabolic chaos, another fundamental system in your body is undergoing its own quiet transformation. Your immune system, that vast network of cells, tissues, and chemical signals that protects you from infection and disease, responds to sleep deprivation with changes that are both immediate and measurable. The immune system isn't a single entity, but rather a collection of interconnected defenses. The innate immune system provides rapid, non-specific responses to threats, while the adaptive immune system develops targeted responses to specific pathogens and remembers them for future encounters. Both branches of immunity show alterations after inadequate sleep, though the effects manifest differently across various immune components. Let's start with one of the most striking findings. the effect of sleep deprivation on natural killer cells. Natural killer cells, often abbreviated as NK cells, represent a crucial component of your innate immune defense. These cells circulate through your bloodstream, constantly surveilling for infected or cancerous cells. When they encounter such cells, NK cells release cytotoxic compounds that destroy the threat. Research examining NK cell activity after sleep restriction has found consistent and dramatic effects. One frequently cited study restricting sleep to just 4 hours for a single night found that NK cell activity decreased to an average of 72% of normal levels. That's a 28% reduction in the functional capacity of these critical immune defenders after just 1 night of partial sleep loss. The implications of reduced NK cell function extend beyond immediate infection risk. NK cells play important roles in controlling viral infections, preventing tumor growth, and regulating immune responses. Long-term epidemiological studies have found associations between reduced NK cell function and increased mortality from cancer over extended follow-up periods. While 1 night of reduced NK activity doesn't equate to cancer risk, it does illustrate how fundamental sleep is to maintaining these protective immune functions. White blood cell counts also respond to sleep deprivation, though the direction of change isn't always what you might expect. Some studies report increases in total white blood cell counts after sleep loss, particularly in specific subsets like neutrophils and granulocytes. These cells, part of the innate immune system's first line of defense, may increase as part of a stress response to sleep deprivation. This increase might seem beneficial. More immune cells should mean better protection, right? Not necessarily. The issue isn't just the number of immune cells, but their function and the overall balance of the immune response. An increase in certain white blood cells accompanied by decreased functional capacity of others, like NK cells, represents dysregulation rather than enhancement. Moreover, the pattern of white blood cell changes after sleep deprivation resembles what's seen during physical or psychological stress, suggesting that the immune system is responding to sleep loss as a physiological stressor. This stress response includes activation of the sympathetic nervous system and increases in stress hormones like cortisol, both of which have complex effects on immune function. Inflammatory markers, chemical signals that coordinate immune responses and indicate the presence of inflammation, show consistent elevation after sleep deprivation. C-reactive protein, a marker of systemic inflammation used clinically to assess cardiovascular and infectious disease risk, increases after sleep loss. Interleukin-6, tumor necrosis factor alpha, and other pro-inflammatory cytokines also show elevations in multiple studies. This inflammatory response isn't necessarily protective inflammation directed at specific threats. Instead, it appears to represent a state of low-grade systemic inflammation, a kind of background noise in the immune system that, when prolonged, contributes to numerous chronic diseases. Inflammation drives atherosclerosis, worsens insulin resistance, promotes neurodegeneration, and accelerates aging-related decline across multiple organ systems. The timing and pattern of immune changes matter significantly. During normal sleep, particularly during the deeper stages of non-REM sleep, your immune system shows specific activation patterns. Certain pro-inflammatory cytokines increase during early sleep, supporting what's called the Th1 response, a branch of adaptive immunity important for fighting intracellular pathogens and supporting long-term immune memory formation. Simultaneously, anti-inflammatory signals also operate during sleep, creating a carefully orchestrated balance. This balance allows beneficial immune activities to occur, such as memory consolidation and tissue repair, without causing excessive inflammation. After sleep deprivation, this delicate balance becomes disrupted. Pro-inflammatory signals may increase without adequate anti-inflammatory counterbalancing, or the normal rhythms of immune activity become desynchronized. Studies examining immune responses to vaccination provide particularly clear evidence of sleep's importance for adaptive immunity. When healthy individuals receive vaccinations after being sleep-deprived, their antibody responses are significantly blunted compared to well-rested individuals. One study found that restricting sleep to 4 hours per night for 6 nights before influenza vaccination resulted in a greater than 50% reduction in antibody production compared to control subjects with normal sleep. This finding has direct practical relevance. If you're planning to get vaccinated, ensuring adequate sleep in the nights before and after vaccination may significantly improve your immune system's ability to develop protective immunity. The investment in sleep directly enhances the vaccine's effectiveness. The mechanisms underlying these immune changes involve multiple pathways. During sleep, particularly deep, slow-wave sleep, the balance of hormones shifts in ways that support immune function. Growth hormone levels surge during early deep sleep. Prolactin and other hormones that support Th1 immune responses also increase. Cortisol, which has immunosuppressive effects, reaches its lowest levels during early sleep before beginning to rise toward morning. This hormonal environment during sleep appears optimal for immune system maintenance and enhancement. Sleep deprivation disrupts these hormonal patterns, potentially explaining many of the observed immune changes. Extended waking means extended exposure to the sympathetic nervous system activation and cortisol patterns associated with being awake, which may suppress certain immune functions while leaving others unchecked. Adenosine, the neuromodulator we discussed earlier in the context of brain function, also affects immune cells. Adenosine receptors exist on various immune cells, and adenosine signaling influences immune cell trafficking, activation, and function. The elevated adenosine levels during sleep deprivation may directly impact immune cell behavior, though the complexity of these interactions is still being unraveled. The circadian clock, your body's internal 24-hour timing system, also regulates immune function independently of sleep itself. Many immune cells carry their own circadian clocks, rhythmic gene expression patterns that cause immune function to vary across the day-night cycle. Sleep deprivation, especially when it involves staying awake during the normal sleep period, creates a mismatch between the circadian system's expectations and actual behavior, potentially contributing to immune dysregulation. Interestingly, not all immune functions are equally affected by acute sleep deprivation. Some aspects of immune function show remarkable resilience to short-term sleep loss. The ability to mount basic inflammatory responses to injuries or infections appears relatively preserved after one night of poor sleep. Your immune system doesn't simply shut down. Rather, specific functions become impaired while others continue operating, creating an altered immune landscape rather than complete immune failure. Individual vulnerability to immune effects of sleep deprivation varies substantially. Age influences the immune response to sleep loss, with some research suggesting that younger individuals may show different patterns than older adults. Physical fitness, baseline health status, stress levels, and prior sleep history all appear to modulate how significantly the immune system responds to acute sleep deprivation. One particularly concerning aspect of these immune changes involves their potential persistence. Some research suggests that immune system alterations induced by sleep deprivation may not immediately resolve with recovery sleep. Studies examining markers of immune cell programming have found that sleep restriction can induce changes in immune stem cells, the precursors that give rise to mature immune cells, and these changes may persist for extended periods even after sleep normalizes. This suggests a kind of immune memory of sleep loss, where brief periods of deprivation leave lasting footprints in immune system function. While more research is needed to fully understand this phenomenon and its clinical significance, it underscores that sleep's influence on immunity extends beyond the immediate period of sleep loss. The clinical implications of these immune changes, even from acute sleep deprivation, shouldn't be underestimated. If you're fighting off a cold, dealing with an infection, or recovering from an injury, sleep becomes even more critical. Your body's ability to mount effective immune responses and complete healing processes depends on adequate sleep. The common wisdom to rest when you're sick has solid biological grounding in the immune impairments associated with sleep deprivation. For individuals with autoimmune conditions, where the immune system attacks the body's own tissues, sleep disruption may worsen disease activity. The inflammatory changes and immune dysregulation associated with sleep loss could exacerbate the underlying immune dysfunction driving autoimmune diseases. While one night of poor sleep is unlikely to trigger a disease flare, it represents one more stressor on an already challenged immune system. Understanding these immune effects provides yet another reason to prioritize sleep. The immune system operates largely outside conscious awareness, quietly protecting you from countless threats every day. Sleep deprivation compromises this protection in ways you don't immediately notice until you fall ill, heal more slowly, or develop chronic inflammation-related health problems. As we move forward to examine cardiovascular effects, keep in mind the interconnections. The inflammatory markers elevated by sleep deprivation directly affect blood vessel health. The stress hormone changes influence blood pressure. The sympathetic nervous system activation affects heart function. Once again, we see that sleep deprivation doesn't affect isolated systems, but rather creates cascading changes across the integrated network of your physiology. Cardiovascular stress. Your heart and blood vessels under pressure. Your cardiovascular system, the intricate network of heart and blood vessels that delivers oxygen and nutrients to every cell in your body while removing waste products, operates under remarkable demands even under normal circumstances. Your heart beats approximately 100,000 times each day, pumping thousands of gallons of blood through 60,000 miles of blood vessels. This system never rests, never takes a break, and yet it maintains remarkable stability across varying conditions. Sleep provides a crucial recovery period for this tireless system. During normal sleep, particularly during the deeper stages of non-REM sleep, your cardiovascular system experiences what amounts to a nightly Sabbath. Heart rate slows. Blood pressure drops by 10 to 20% in a phenomenon called nocturnal dipping. The workload on your heart decreases, allowing cardiovascular tissues to recover from the day's demands. When sleep is disrupted or insufficient, this cardiovascular reprieve is shortened or eliminated, and the consequences manifest quickly. Even after just one night of inadequate sleep, measurable changes occur in cardiovascular function, changes that, while temporary, mirror patterns seen in cardiovascular disease. Let's begin with blood pressure, one of the most fundamental cardiovascular parameters. Under normal circumstances, blood pressure follows a predictable daily rhythm. It's lowest during sleep, rises in the morning, remains relatively elevated during waking hours, then dips again at night. This nocturnal dipping appears to be physiologically important, giving the cardiovascular system a period of reduced mechanical stress. Studies examining blood pressure during and after sleep deprivation consistently find alterations in this pattern. When you stay awake during your normal sleep period, blood pressure doesn't show the expected nocturnal dip. Instead, it remains elevated throughout the night. This non-dipping pattern has been associated with increased cardiovascular disease risk in epidemiological studies. People whose blood pressure fails to dip adequately at night show higher rates of stroke, heart attack, and kidney disease. The morning after sleep deprivation, blood pressure often remains elevated compared to normal. Studies measuring blood pressure across the day following inadequate sleep show increases in both systolic pressure, the top number reflecting the pressure when your heart contracts, and diastolic pressure, the bottom number reflecting pressure between heartbeats. These increases might seem modest in absolute terms, perhaps 5 to 10 millimeters of mercury, but they represent measurable additional stress on the cardiovascular system. The mechanisms driving these blood pressure changes involve multiple factors. Sympathetic nervous system activity, the branch of your autonomic nervous system responsible for the fight or flight response, increases during sleep deprivation. This sympathetic activation causes blood vessels to constrict, increasing resistance to blood flow and thereby raising blood pressure. It also increases heart rate and the force of heart contractions, further elevating blood pressure. Cortisol and other stress hormones, which we've discussed in previous sections, also contribute to blood pressure elevation. Cortisol affects kidney function, altering how your body handles sodium and water, which influences blood volume and blood pressure. The disrupted cortisol patterns seen after sleep deprivation may thus contribute to the observed blood pressure changes. Interestingly, some research finds that while blood pressure increases, muscle sympathetic nerve activity, a direct measure of sympathetic signals going to blood vessels, may actually decrease in some individuals after sleep deprivation. This seemingly paradoxical finding suggests that the blood pressure elevation might result more from changes in blood vessel responsiveness or blood volume than from increased sympathetic drive, per se. The cardiovascular system's response to sleep deprivation appears more complex than simple sympathetic activation. Heart rate variability, a measure of the variation in time intervals between heartbeats, provides another window into cardiovascular function and autonomic nervous system balance. Higher heart rate variability generally indicates better cardiovascular health and greater autonomic flexibility. The heart can speed up and slow down in response to changing demands, reflecting responsive and adaptable cardiovascular control. Sleep deprivation consistently reduces heart rate variability, indicating reduced parasympathetic activity, the calming branch of the autonomic nervous system, and a shift towards sympathetic predominance. This autonomic imbalance, with too much sympathetic activation and insufficient parasympathetic counterbalancing, appears to be a consistent feature of the cardiovascular response to inadequate sleep. The reduction in heart rate variability isn't merely a statistical curiosity. Lower heart rate variability has been associated with increased risk of cardiovascular events and mortality in numerous studies. While one night of reduced heart rate variability doesn't equate to long-term cardiovascular risk, it demonstrates that sleep deprivation creates cardiovascular conditions resembling those seen in cardiovascular disease. Endothelial function, referring to the health and responsiveness of the cells lining your blood vessels, also shows impairment after sleep deprivation. The endothelium does far more than simply provide a physical barrier. It actively regulates blood vessel tone, inflammation, and clotting. Healthy endothelium produces nitric oxide, a molecule that causes blood vessels to relax and dilate, improving blood flow and reducing blood pressure. Studies examining endothelial function after sleep deprivation, typically using techniques that measure how well blood vessels dilate in response to increased blood flow, find reduced endothelial responsiveness. Blood vessels don't dilate as effectively as they should. This endothelial dysfunction represents an early step in the development of atherosclerosis, the buildup of plaques in blood vessels that underlies heart attacks and strokes. The mechanisms driving endothelial dysfunction after sleep loss likely involve the inflammatory changes we discussed in the previous section. Inflammatory cytokines and oxidative stress impair endothelial cells' ability to produce nitric oxide. The sympathetic activation and altered hormonal patterns may also directly affect endothelial function. Whatever the precise mechanisms, the result is measurably impaired blood vessel health after even brief sleep deprivation. Circulating markers of endothelial activation and damage also increase after sleep loss. Molecules like E-selectin and ICAM1, adhesion molecules that increase when endothelial cells are activated or damaged, show elevations following sleep deprivation. These markers are used clinically to assess cardiovascular disease risk, and their elevation after sleep loss suggests that acute sleep deprivation creates a cardiovascular environment resembling early disease states. Studies examining the response to acute stressors after sleep deprivation reveal additional cardiovascular vulnerabilities. When sleep-deprived individuals undergo stressful tasks, their blood pressure and heart rate responses to those stressors are often exaggerated compared to rested states. The cardiovascular system overreacts to challenges, showing larger increases in blood pressure and requiring longer recovery times to return to baseline. This exaggerated stress reactivity has important practical implications. If you're sleep-deprived and encounter a stressful situation, whether it's a tense meeting, a near miss while driving, or an argument, your cardiovascular system may respond more intensely than it otherwise would. These repeated, exaggerated cardiovascular responses to daily stressors, if they occur chronically due to ongoing sleep deprivation, could contribute to cardiovascular disease development over time. Cardiac rhythm disturbances represent another potential concern. While healthy young adults typically don't develop serious arrhythmias from one night of poor sleep, sleep deprivation can affect cardiac electrical activity in measurable ways. Changes in autonomic balance, electrolyte shifts, and direct effects on cardiac muscle cells could theoretically predispose to rhythm problems, particularly in individuals with underlying cardiac conditions. Some research examining athletes and individuals undergoing extreme sleep deprivation has documented increased frequency of premature ventricular contractions, extra heartbeats originating from the heart's lower chambers. While these are often benign, they represent electrical instability. In vulnerable individuals, more serious rhythm disturbances could potentially occur. The timing of cardiovascular events follows a circadian pattern, with heart attacks and strokes occurring more frequently in the morning hours shortly after waking. The mechanisms underlying this morning vulnerability include the surge in sympathetic activity upon waking, the morning rise in blood pressure, increased blood clotting tendency in morning hours, and the cardiovascular demands of transitioning from sleep to active waking. Sleep deprivation appears to amplify some of these morning cardiovascular stresses. The blood pressure surge may be more pronounced, the sympathetic activation more intense, and the overall cardiovascular strain greater. While one morning after poor sleep is unlikely to trigger a cardiac event in healthy individuals, for those with existing cardiovascular disease, the additional stress could represent a meaningful increase in risk. Chronic effects of repeated sleep deprivation on cardiovascular health are increasingly well documented in epidemiological research. People who consistently sleep less than 6 hours per night show higher rates of hypertension, coronary artery disease, heart failure, stroke, and cardiovascular mortality compared to those sleeping 7 to 8 hours. The mechanisms we've been discussing, blood pressure elevation, endothelial dysfunction, inflammation, autonomic imbalance, likely contribute to these long-term cardiovascular consequences. What's particularly striking is how quickly the acute cardiovascular changes occur. You don't need months or years of poor sleep to see blood pressure rise, heart rate variability decrease, or endothelial function decline. These changes happen within hours to days, demonstrating how fundamental sleep is to cardiovascular health. Individual vulnerability to cardiovascular effects of sleep deprivation varies. Age is a significant factor, with older individuals potentially showing greater cardiovascular responses to sleep loss. Pre-existing cardiovascular conditions, such as hypertension or coronary disease, increase vulnerability. Genetic factors likely play roles as well, with some individuals showing greater cardiovascular sensitivity to sleep disruption than others. Physical fitness may provide some protection. Individuals with better cardiovascular fitness appear to tolerate sleep deprivation somewhat better from a cardiovascular standpoint, though fitness doesn't eliminate the effects entirely. Regular exercise's beneficial effects on endothelial function, autonomic balance, and blood pressure regulation may buffer against some of the cardiovascular stress of sleep loss. The cardiovascular effects of sleep deprivation represent more than abstract physiological changes. They translate directly into increased risk for acute cardiovascular events in vulnerable individuals and contribute to long-term cardiovascular disease development with repeated exposure. Understanding these effects underscores why cardiovascular health organizations increasingly emphasize adequate sleep as a fundamental pillar of cardiovascular disease prevention, alongside traditional factors like diet, exercise, and smoking cessation. As we transition to examining how sleep deprivation affects physical performance and coordination, remember that these cardiovascular changes don't occur in isolation from the other effects we've discussed. The metabolic disruption affects cardiovascular health. The immune activation and inflammation impair blood vessel function. The sympathetic nervous system activation influences both cardiovascular and metabolic systems. Your body's integrated response to sleep deprivation touches every system, and each system's response influences the others. Physical performance, when your body won't cooperate. After examining the internal disruptions occurring in your brain, metabolism, emotions, immune system, and cardiovascular function, you might wonder how all these changes manifest in your physical capabilities. How does one night of poor sleep affect your ability to move, coordinate, perform athletic activities, and execute the physical tasks of daily life? The answer is nuanced and somewhat surprising. Contrary to what you might expect, maximal physical efforts, those requiring you to exert maximum strength or power for brief periods, often show relatively modest impairment after acute sleep deprivation. If you're asked to lift the heaviest weight you can manage, jump as high as possible, or sprint as fast as you can, your performance might decline only slightly or not at all after one night of poor sleep. This resilience of maximal performance appears related to motivation and the ability to briefly override fatigue. When a task is short and you're highly motivated, you can often summon near-normal effort despite being sleep-deprived. Your nervous system can generate forceful muscle contractions, and your cardiovascular system can deliver oxygen for brief intense efforts, even when operating under the stress of sleep loss. Where performance really suffers is in submaximal efforts requiring sustained attention, fine motor control, and coordination. These are the physical activities demanding not just muscular strength, but also precise neuromuscular control. The ability to coordinate muscle activation patterns, maintain balance, and execute complex movement sequences. Studies examining athletic performance after sleep deprivation reveal this pattern clearly. Measures of maximum strength, such as 1 repetition maximum lifts, show minimal impairment after sleep loss. However, performance on tasks requiring sustained effort, like running a set distance at a self-selected pace, shows clear degradation. Athletes run shorter distances, report greater perceived exertion for the same workload, and fatigue more quickly. Time to exhaustion, how long you can maintain a given level of physical effort, consistently decreases after sleep deprivation. Even when maximum power output is preserved, the ability to sustain near maximal efforts diminishes. This appears related to both physiological changes, such as altered substrate metabolism and potentially reduced glycogen stores, and perceptual changes, where effort feels harder and discomfort feels more intense. Perceived exertion, your subjective sense of how hard you're working, increases dramatically after sleep deprivation. The same physical effort that felt moderate when well-rested feels strenuous when sleep-deprived. This heightened perception of effort likely contributes to reduced endurance performance. When exercise feels more uncomfortable, motivation to continue wanes and performance suffers. The mechanisms underlying this exaggerated perceived exertion aren't entirely clear, but they likely involve the same brain regions affected by sleep deprivation that we've discussed in earlier sections. The prefrontal cortex, involved in evaluating sensations and making decisions about continuing or stopping effortful activities, operates suboptimally after sleep loss. The altered emotional processing we discussed may make discomfort feel more aversive. The metabolic changes may alter the actual physiological strain of exercise. Skill-based performance shows particularly pronounced impairment after sleep deprivation. Activities requiring precise timing, accuracy, and coordination suffer more than pure strength or power tasks. Tennis serving accuracy declines. Basketball free throw percentage drops. Fine motor control in tasks like threading needles or assembling small parts deteriorates. Research examining skill control in athletes finds effect sizes suggesting substantial performance degradation. Sleep-deprived athletes show reduced serving accuracy in volleyball, decreased shooting percentage in basketball, and impaired performance in skill-based drills across various sports. The decline in skill performance often exceeds the decline in physical capacities like strength or speed. These skill impairments make biological sense, given what we know about sleep deprivation's effects on the brain. Skill execution requires precise timing of muscle activation, integration of sensory feedback, and real-time adjustment based on performance monitoring. All of these processes depend on brain regions we know are affected by sleep loss, particularly the prefrontal cortex, cerebellum, and motor cortex. Reaction time, critical for many sports and daily activities, slows measurably after sleep deprivation. Simple reaction time, responding to a single stimulus, shows moderate slowing. Choice reaction time, requiring you to make decisions about which response is appropriate, shows greater impairment. The more complex the decision required, the greater the performance decline. This reaction time slowing directly impacts performance in sports requiring rapid responses, such as baseball batting, where milliseconds determine whether you make contact with the ball. It also affects driving safety, where delayed reactions to braking vehicles or unexpected hazards increase accident risk. The slowing may seem modest in laboratory measurements, but in real-world contexts where timing is critical, even small delays can have significant consequences. Balance and postural control represent another domain of physical performance affected by sleep deprivation. Maintaining balance requires integration of multiple sensory inputs, visual information, vestibular signals from your inner ear, and proprioceptive feedback from muscles and joints, along with motor output to make corrective adjustments. Sleep deprivation appears to impair this sensory integration and motor control. Studies measuring postural sway, the subtle movements your body makes to maintain balance, find increased sway after sleep deprivation, particularly in challenging conditions like standing on one leg or with eyes closed. The increased sway indicates less stable balance control. For most people in most situations, this doesn't result in falling, but it represents degraded neuromuscular control that could matter in demanding circumstances. Motor learning, your ability to acquire new motor skills or refine existing ones, appears to depend critically on sleep, particularly the sleep-following practice. Research shows that motor skill consolidation, The process by which a newly practiced skill becomes more stable and automatic occurs primarily during sleep. Specific brain regions show reactivation during sleep of patterns that were active during motor learning, suggesting that the brain is replaying and strengthening motor memories. When sleep is disrupted or insufficient after motor practice, this consolidation process is impaired. Athletes learning new techniques, musicians practicing new pieces, or anyone acquiring new motor skills will show reduced improvement after sleep-deprived nights compared to nights with adequate sleep. The practice itself may occur normally, but the offline consolidation that normally happens during sleep is compromised. Coordination between multiple body parts also suffers under sleep deprivation. Tasks requiring bilateral coordination using both sides of the body in synchronized or complementary ways show impairment. The temporal precision of movement sequences decreases. You're more likely to make timing errors, to have movements feel awkward or uncoordinated. This coordination impairment likely reflects the same prefrontal and motor cortex dysfunction underlying other motor performance deficits. The brain regions responsible for planning, initiating, and monitoring complex movement sequences operate less efficiently after sleep loss, and the quality of movement control reflects this inefficiency. Exercise-induced fatigue, the normal tiredness that develops during physical activity, appears to develop more quickly and more intensely when you're sleep-deprived. Studies examining the physiological responses to exercise find that some parameters like heart rate and lactate accumulation may be elevated at given exercise intensities after sleep deprivation, suggesting that the same workload creates greater physiological strain. However, some studies find minimal changes in these physiological markers, suggesting that the increased perceived exertion and reduced performance may stem more from central factors, changes in the brain and nervous system, than from peripheral factors in the muscles themselves. The muscles may be capable of producing force and sustaining activity, but the nervous system driving them and the brain evaluating the experience of exertion are compromised. Injury risk represents a particularly concerning potential consequence of sleep-deprived physical performance. While direct evidence linking acute sleep deprivation to increased injury occurrence is limited, the mechanisms are plausible. Impaired coordination, slower reaction times, degraded balance, and altered movement patterns all theoretically increase injury vulnerability. Studies in athletes have found associations between chronic sleep restriction and increased injury rates, suggesting that inadequate sleep does indeed compromise the body's ability to safely execute demanding physical activities. Whether a single night of poor sleep meaningfully increases injury risk likely depends on the activity's demands and the individual's baseline capabilities. But the theoretical mechanisms suggest caution is warranted. Recovery from exercise also appears to be affected by sleep quality. Sleep is when your body undertakes many of the repair and improvement processes that allow you to recover from training and become fitter. Growth hormone secretion during deep sleep supports tissue repair. Protein synthesis increases during sleep. Glycogen stores, the carbohydrate fuel stored in muscles, replenish during rest periods. When sleep is inadequate, these recovery processes are truncated. Studies examining recovery markers like muscle glycogen have found that sleep-deprived athletes show reduced glycogen restoration compared to well-rested counterparts. This means that even if you can perform adequately today after poor sleep, tomorrow's performance may suffer because your body hasn't fully recovered from yesterday's efforts. The autonomic nervous system imbalance we discussed in the cardiovascular section also affects recovery. Proper recovery requires parasympathetic nervous system dominance, the rest and digest state. Sleep deprivation's shift toward sympathetic predominance interferes with this recovery state. Your body remains in a more activated, stressed state even during supposed rest periods, impairing recovery quality. Individual variability in physical performance responses to sleep deprivation is substantial. Some athletes show dramatic performance decrements after poor sleep, while others maintain near-normal performance. Factors like training status, motivation, the specific demands of the activity, and perhaps genetic factors all appear to influence vulnerability to performance impairment. Interestingly, the time of day when performance is assessed relative to sleep deprivation matters. Performance tends to be worse later in the day following sleep deprivation compared to morning testing. This time since awakening effect suggests that cumulative time awake contributes to performance degradation beyond the sleep loss itself. The practical implications for anyone engaged in physical activities are clear. On days after poor sleep, extra caution is warranted for activities requiring precise coordination, sustained effort, or rapid reactions. Adjusting training intensity, avoiding particularly risky activities, or simply being aware of impairment can help mitigate the consequences of sleep-deprived physical performance. For competitive athletes, the message is unambiguous. Sleep is a performance intervention as important as training and nutrition. The athletes who sleep adequately perform better, learn motor skills more effectively, and recover more completely than those who sacrifice sleep. No amount of additional training can compensate for inadequate sleep when it comes to optimizing performance. As we move into examining the specific brain mechanisms underlying all the effects we've been discussing, the physical performance impairments we've explored will make even more sense. The coordination problems, reaction time slowing, and altered perceived exertion all stem from the same neural changes affecting cognition and emotion. Your brain, operating under the stress of accumulated adenosine, hormonal disruption, and cellular fatigue, simply can't coordinate your body's movements with its usual precision and efficiency. The brain under duress, neural mechanisms, and microsleeps. Throughout our exploration of sleep deprivation's effects, we've touched on various brain changes underlying the cognitive, emotional, metabolic, and physical consequences. Now let's examine the brain's response to sleep loss more directly, understanding the specific neural mechanisms that drive everything we've discussed. At the most fundamental level, sleep deprivation represents a mismatch between your brain's biological needs and its actual state. Your brain needs sleep for multiple reasons: to clear metabolic waste products, to restore energy supplies, to consolidate memories, to recalibrate neural circuits. When sleep doesn't occur, these needs go unmet, and compensatory processes begin. Perhaps the most important molecule in understanding sleep deprivation is adenosine. Adenosine is a neuromodulator, a chemical that modulates neuronal activity, and its accumulation during waking appears to be one of the primary drivers of what researchers call sleep pressure, the biological urge to sleep. Adenosine is produced as a byproduct of cellular energy metabolism. When neurons are active, they consume ATP, the cellular energy currency, breaking it down to ADP and eventually to adenosine. During waking, when neurons throughout your brain are active, adenosine steadily accumulates in the extracellular space, the fluid surrounding brain cells. This accumulating adenosine has inhibitory effects on neural activity. It binds to adenosine receptors, particularly A1 receptors, on neurons in various brain regions. When adenosine binds to these receptors, it reduces neuronal excitability and decreases neurotransmitter release. The overall effect is a damping down of neural activity, a biological signal indicating that the brain needs rest. During sleep, particularly deep non-REM sleep, adenosine levels decrease. The mechanisms aren't entirely clear, but reduced neural activity during sleep means less ATP consumption and less adenosine production. Additionally, cellular processes that clear adenosine may operate more effectively during sleep. By morning, after a full night's sleep, adenosine levels have returned to low baseline values, and you wake refreshed with sleep pressure reset to 0. After sleep deprivation, adenosine levels remain elevated or increase beyond normal waking levels. Studies measuring adenosine in various brain regions have found particularly consistent increases in the basal forebrain, a region at the base of the front of the brain that plays crucial roles in arousal and attention. This elevated adenosine in the basal forebrain appears especially important because this region contains neurons that promote wakefulness and are critical for maintaining cortical activation. When adenosine accumulates to high levels in the basal forebrain, it inhibits these wake-promoting neurons. The result is that even though you're technically awake, the neural systems that maintain full wakefulness are operating under heavy inhibitory pressure. Your brain exists in a kind of twilight state, neither fully awake nor fully asleep. This adenosine accumulation helps explain many of the cognitive impairments we've discussed. The prefrontal cortex appears particularly sensitive to sleep deprivation, and adenosine effects in prefrontal regions likely contribute to impaired executive function, attention, and working memory. The inability to sustain attention, the increased susceptibility to distraction, the difficulty with complex cognitive tasks all reflect prefrontal cortex operating under adenosine's inhibitory influence. But adenosine doesn't act alone. Its effects interact with other neuromodulatory systems. Acetylcholine, a neurotransmitter crucial for attention, learning, and cortical activation, shows reduced release after sleep deprivation, partly due to adenosine's inhibitory effects on cholinergic neurons in the basal forebrain. This acetylcholine reduction contributes to the attentional impairments and learning difficulties seen with sleep loss. Dopamine, a neurotransmitter involved in motivation, reward, and motor control, also shows changes after sleep deprivation. Studies examining dopamine receptors have found that sleep deprivation leads to downregulation of D2 and D3 dopamine receptors in certain brain regions, particularly the striatum. This receptor downregulation may contribute to the altered reward sensitivity, impaired motivation, and modified risk-taking we've discussed. The relationship between adenosine and dopamine is particularly interesting. Adenosine and dopamine receptors can physically interact, forming heteromers, where activation of adenosine receptors can reduce dopamine receptor function. The elevated adenosine during sleep deprivation may thus directly impair dopamine signaling, contributing to motivational and reward-related changes. Brain imaging studies using functional MRI provide a more macroscopic view of how sleep deprivation affects brain activity. Consistent findings include reduced activation in the dorsolateral prefrontal cortex, reduced activity in the intraparietal sulcus, and altered activity in the thalamus during cognitive tasks performed while sleep-deprived. These regions show less vigorous responses to cognitive demands, suggesting they're operating less efficiently. Interestingly, some brain regions show increased activation after sleep deprivation, possibly representing compensatory responses where the brain attempts to recruit additional resources to maintain performance. The extent and location of these compensatory activations vary across individuals and tasks, and they're typically insufficient to fully preserve normal performance. Connectivity analyses examining how different brain regions communicate with each other reveal disrupted functional connectivity after sleep deprivation. The default mode network, a set of brain regions active during rest and mind wandering, shows altered connectivity. The frontoparietal attention network shows reduced connectivity. And as we discussed in the emotional regulation section, connectivity between the prefrontal cortex and amygdala becomes disrupted. These connectivity changes suggest that sleep deprivation doesn't just affect individual brain regions in isolation, but rather disrupts the coordinated activity of brain networks. The brain becomes less integrated, with reduced communication between regions that need to work together for complex cognitive functions. Electrical brain activity, measured using EEG, shows characteristic changes during sleep deprivation. The predominant frequency of waking brain activity slows, with more theta wave activity, typically associated with drowsiness, appearing during intended wakefulness. This EEG slowing reflects the drowsy, semi-awake state of the sleep-deprived brain. During tasks requiring attention, the brain's event-related potentials, electrical responses to specific stimuli, show alterations after sleep deprivation. The P300 component, a positive-going electrical wave occurring about 300 milliseconds after a stimulus that reflects attention and working memory processes, shows increased latency and sometimes reduced amplitude after sleep loss. This suggests slower and less efficient attentional processing. One of the most fascinating and concerning phenomena of sleep deprivation is the occurrence of microsleeps, brief episodes where you essentially fall asleep for a fraction of a second to several seconds, even while ostensibly awake and engaged in activity. During microsleeps, your brain waves briefly show sleep-like patterns. Your behavioral responsiveness ceases, and if your eyes are being monitored, they may show slow rolling movements or partial closure. Microsleeps appear to represent local sleep intrusion into wakefulness. Studies using high-density EEG, which can measure brain activity with fine spatial resolution, suggest that during microsleeps, certain brain regions enter sleep states, while others remain awake. Your brain becomes a mosaic of sleeping and waking regions rather than a uniformly awake organ. The regions most likely to enter local sleep states during microsleeps appear to be those under the heaviest adenosine burden or those most crucial for the tasks you're attempting. If you're trying to maintain visual attention, occipital visual cortex regions might show sleep-like activity. If you're performing a motor task, motor cortex regions might briefly sleep. These microsleeps are particularly dangerous because they occur without warning and without your awareness. You might experience a microsleep while driving, during which time you're completely unresponsive to road conditions, yet you may have no memory of having lost consciousness. Your last conscious memory is of driving normally. Then suddenly several seconds have passed that you can't account for. Crashes, errors, and accidents can occur during these brief lapses. The frequency of microsleeps increases with greater sleep deprivation and longer time awake. After moderate sleep restriction, you might experience only occasional microsleeps. After more severe deprivation or very extended waking, microsleeps can occur dozens of times per hour. Any activity requiring sustained attention becomes essentially impossible when microsleeps are frequent. Neurochemically, sleep deprivation affects numerous neurotransmitter systems beyond adenosine, dopamine, and acetylcholine. Serotonin, involved in mood regulation, shows alterations. Norepinephrine, important for arousal and stress responses, is affected. GABA, the primary inhibitory neurotransmitter, and glutamate, the primary excitatory neurotransmitter, show changes in their balance. The brain's entire neurochemical landscape shifts in response to inadequate sleep. Cellular stress responses activate after sleep deprivation. Studies examining markers of cellular stress, like heat shock proteins and inflammatory signals within brain cells, find evidence that neurons are experiencing stress conditions after extended waking. This cellular stress might contribute to the cognitive impairments and could have implications for long-term brain health if sleep deprivation becomes chronic. The glymphatic system, a recently discovered brain waste clearance system that operates primarily during sleep, provides another mechanism linking sleep to brain function. During sleep, research suggests the space between brain cells expands, and cerebrospinal fluid flows through brain tissue, carrying away metabolic waste products, including potentially toxic proteins like beta-amyloid. When sleep is disrupted, glymphatic function appears to be impaired, and waste clearance is reduced. Studies have found that even one night of sleep deprivation can lead to increased beta-amyloid in the brain, a protein that accumulates in Alzheimer's disease. While one night of elevated beta-amyloid doesn't cause Alzheimer's, these findings demonstrate that sleep loss impairs this crucial waste clearance process. Synaptic homeostasis, the regulation of synaptic strength throughout the brain, also depends on sleep. During waking, synapses, the connections between neurons, tend to strengthen as you learn and experience the world. If this strengthening continued indefinitely, synapses would become saturated, unable to encode new information. Sleep appears to provide a period of synaptic downscaling, where synapses are pruned and weakened in a coordinated way that preserves important information while making room for new learning. Sleep deprivation appears to disrupt this synaptic homeostasis. Research suggests that synapses don't undergo the normal downscaling, which could lead to cognitive inefficiency and impaired ability to learn new information. The brain becomes cluttered with unnecessary synaptic connections while lacking the synaptic plasticity needed for new memory formation. Regional differences in vulnerability to sleep deprivation are important to understand. The prefrontal cortex consistently emerges as particularly sensitive to sleep loss across multiple types of studies. This sensitivity might reflect the high metabolic demands of prefrontal regions, their dense adenosine receptor expression, or the complexity of the computations they perform. Whatever the reasons, prefrontal vulnerability helps explain why executive functions, emotional regulation, and decision-making are so notably impaired by sleep deprivation. The thalamus, a central relay station for sensory information, also shows functional changes after sleep deprivation. Reduced thalamic activity could impair the transmission of sensory information to cortical processing regions, contributing to attentional deficits and perceptual impairments. Brain metabolism, the consumption of glucose and oxygen by brain tissue, shows alterations after sleep deprivation. Overall, cerebral glucose metabolism may decrease in certain brain regions, particularly the prefrontal cortex and thalamus, reflecting reduced neural activity in these areas. This metabolic decline accompanies and likely contributes to the functional impairments we've been discussing. Understanding these neural mechanisms helps integrate all the effects we've explored into a coherent picture. The cognitive fog stems from prefrontal cortex dysfunction driven by adenosine accumulation and altered neurotransmitter balance. The emotional volatility reflects amygdala hyperreactivity combined with reduced prefrontal control, both consequences of the same neural disruptions. The physical performance impairments reflect motor cortex and cerebellar dysfunction under the same pressures. It all traces back to a brain operating under biological conditions outside its normal parameters. Sleep deprivation represents a fundamental violation of your brain's needs, and the neural changes we've examined are your brain's best attempt to function under these adverse circumstances. The remarkable thing isn't how impaired you become after sleep loss. It's that your brain manages to function at all, given the magnitude of neurochemical and metabolic disruption occurring. As we transition to discussing recovery, understanding these neural mechanisms becomes crucial. Recovery isn't just about feeling less tired. It's about allowing adenosine to clear, neurotransmitter systems to rebalance, synapses to undergo proper homeostatic regulation, waste products to be cleared, and normal patterns of brain activity and connectivity to be restored. The brain has substantial work to do during recovery sleep, and understanding this work helps us appreciate why recovery takes time, and why adequate sleep can't be treated as optional. The road to recovery, timeline, and what actually helps. After exploring all the ways that one bad night affects your body and brain, you might be wondering, how long does it take to recover? Can you truly bounce back, or do the effects linger? And what actually helps speed recovery? The answers to these questions are both reassuring and sobering. The reassuring news: acute sleep deprivation is recoverable. Your body possesses remarkable resilience, and given adequate opportunity, most of the effects we've discussed will resolve. The sobering news: recovery takes longer than most people expect, and different functions recover at different rates. Let's start with what happens when you finally get sleep after a night of deprivation. Recovery sleep looks different from normal sleep. After sleep deprivation, your body prioritizes certain sleep stages, particularly deep, slow-wave sleep. The first recovery night typically shows a substantial increase in deep sleep, sometimes called slow-wave rebound. Your brain enters these deep sleep stages more quickly, stays in them longer, and the slow waves themselves may have greater amplitude. This prioritization makes biological sense. Deep sleep appears to be when many of the restorative processes we've discussed occur most intensively. Adenosine clearance, synaptic homeostasis, glymphatic waste clearance, and metabolic restoration all appear to occur preferentially during deep sleep. Your brain, in its wisdom, emphasizes the sleep stage most needed for recovery. REM sleep, the stage associated with dreaming, may also show rebound effects, though sometimes not until the second or third recovery night. This suggests a hierarchy of sleep needs, with deep sleep being most urgent, followed by REM sleep once deep sleep debt has been partially addressed. The subjective experience of recovery varies among individuals, but typical patterns emerge. After one recovery night, you'll likely feel substantially better than you did during sleep deprivation. The overwhelming sleepiness lifts. You feel more alert. Basic cognitive functions improve noticeably. Many people report feeling essentially normal after one good night's sleep following acute deprivation. However, objective testing reveals a more nuanced picture. While some functions recover quickly, others lag behind. Research studies tracking recovery after sleep restriction show that different cognitive and physiological measures return to baseline at different rates. Reaction time, one of the most sensitive measures of sleep deprivation, often shows substantial improvement within 24 hours of recovery sleep. After one full night of adequate sleep, reaction times may return to near baseline levels. This relatively rapid recovery helps explain why people feel cognitively restored even when other deficits remain. Attention and vigilance also recover relatively quickly, though not always completely after just one recovery night. The frequency of attentional lapses decreases dramatically. The ability to sustain attention improves notably. For practical purposes, attention may feel essentially normal after adequate recovery sleep, even if subtle impairments remain detectable with sensitive testing. More complex cognitive functions show slower recovery. Working memory capacity may require multiple nights to fully normalize. Executive function tasks, particularly those involving cognitive flexibility and inhibitory control, might show residual impairment even after a week of recovery sleep. Some research suggests that accuracy on complex cognitive tasks, as opposed to speed, recovers more slowly than simple measures like reaction time. These findings suggest a dissociation between feeling recovered and being fully recovered. You might feel alert and functional while still showing measurable deficits in complex cognitive tasks. This perception-performance gap has important practical implications. You might return to demanding cognitive work after one recovery night feeling capable, but your actual performance on complex problems might still be subtly impaired. Emotional regulation appears to improve relatively quickly with recovery sleep. The amygdala hyperreactivity and reduced prefrontal amygdala connectivity documented after sleep deprivation show substantial improvement after adequate sleep. Subjectively, emotional volatility decreases, irritability subsides, and you feel more emotionally stable. This relatively rapid emotional recovery contributes to the overall sense of being restored after recovery sleep. Metabolic parameters show more variable recovery patterns. Some studies find that glucose metabolism begins normalizing within a day or two of recovery sleep. Insulin sensitivity improves, though may not fully return to baseline immediately. The appetite hormone disruptions, the elevated ghrelin and reduced leptin, appear to resolve relatively quickly with adequate sleep, usually within a few days. However, some metabolic effects might persist longer than expected. Research examining chronic sleep restriction followed by recovery periods has found that even after a week of recovery sleep, some metabolic measures haven't fully normalized. This suggests that metabolic systems might have a kind of memory of sleep deprivation, taking longer to fully reset than some other systems. Immune function recovery follows its own timeline. Natural killer cell activity, so dramatically suppressed during sleep deprivation, appears to improve substantially with recovery sleep. Inflammatory markers like C-reactive protein and cytokines generally decrease toward baseline with adequate rest. The immune system's response to vaccines improves when sleep normalizes. Most immune functions likely return to normal within a few days of adequate sleep, though some research suggests lingering effects on immune cell populations. Cardiovascular parameters like blood pressure and heart rate variability show improvement relatively quickly with recovery sleep. Blood pressure decreases, heart rate variability increases, and the normal patterns of nocturnal dipping and diurnal rhythm restore. The endothelial dysfunction and vascular changes documented after sleep loss appear to be reversible with adequate recovery, though the timeline for complete normalization isn't entirely clear. The timeline for physical performance recovery depends partly on what aspect of performance you're measuring. Perceived exertion, the subjective sense of how hard exercise feels, improves dramatically with recovery sleep. You immediately notice that the same activities that felt exhausting yesterday feel manageable today. Coordination, skill execution, and reaction time also show relatively rapid improvement. Measures of endurance and sustained performance may take longer to fully normalize, particularly if the sleep deprivation affected muscle glycogen stores or other metabolic parameters affecting sustained exercise capacity. So, what's the overall timeline for recovery from one bad night? Based on available research, if we're talking about a single night of partial sleep restriction, say 4 to 5 hours instead of 8, most measurable effects appear to largely resolve within 2 to 3 days of adequate sleep. Subjectively, you'll feel substantially better after just one recovery night. Simple cognitive measures like reaction time normalize quickly. More complex functions require a bit longer, but are generally back to baseline within a few days, according to studies tracking recovery patterns. If the sleep deprivation was more severe, a night of total sleep deprivation or multiple nights of restriction, recovery takes proportionately longer. Complete recovery might require a week or more of consistently adequate sleep. And importantly, getting one good night followed by another night of inadequate sleep doesn't allow full recovery. The restoration process requires sustained adequate sleep, night after night, not just sporadic good nights interspersed with continued deprivation. Research examining chronic sleep restriction, multiple nights of inadequate sleep, has found that even after a full week of recovery sleep, some cognitive measures haven't returned to baseline. This suggests that the debt accumulated through repeated sleep loss takes considerable time to fully repay. The old adage about needing one recovery night for each night of missed sleep may actually underestimate the recovery time needed based on these findings. Individual differences in recovery are substantial. Some people bounce back quickly, showing near-complete recovery after 1 or 2 nights of adequate sleep. Others show more persistent impairments, requiring longer recovery periods. Age might matter, with some evidence suggesting younger individuals recover faster than older adults, though findings are mixed. Genetic factors likely play roles, with variations in genes related to sleep regulation, affecting both vulnerability to sleep deprivation and recovery speed. The concept of sleep debt is useful, but potentially misleading. If you miss 2 hours of sleep one night, you might expect to repay that debt by sleeping 2 hours extra the next night. However, research suggests it's not quite that simple. Getting 2 extra hours helps significantly, but may not immediately restore all functions to baseline. The debt seems to involve more than just making up the hours. The quality and structure of sleep during recovery matters, not just the quantity. Strategic napping can aid recovery, though it doesn't fully substitute for nighttime sleep. A 20 to 30-minute nap can provide substantial alertness benefits, reducing sleepiness and improving attention for several hours. Longer naps, an hour or more, can provide more extensive restorative benefits, but risk sleep inertia, grogginess immediately upon waking. Napping too late in the day might interfere with nighttime sleep, delaying full recovery. Caffeine provides temporary relief from sleepiness, but doesn't address the underlying deficits. It works by blocking adenosine receptors, temporarily countering adenosine's sleep-promoting effects. This can improve alertness and attention acutely, but doesn't restore the functions impaired by sleep deprivation. Once caffeine wears off, you often feel even more tired as adenosine binds to the now available receptors. Caffeine is best viewed as a temporary coping strategy, not a recovery tool. Light exposure can help regulate circadian rhythms and promote alertness during recovery. Bright light, especially early in the day, helps reinforce your circadian system and promotes wakefulness when you need it. Avoiding bright light, especially blue-enriched light, in evening hours helps promote sleepiness and quality sleep when recovery time arrives. Physical activity has complex effects on recovery. On one hand, exercise might feel more difficult after sleep deprivation and could add to your body's stress burden. On the other hand, regular exercise generally improves sleep quality and might aid recovery. The appropriate approach likely involves reducing exercise intensity and duration during acute recovery from sleep deprivation, allowing your body to focus resources on restoration rather than demanding further performance. The most important factor in recovery is simply getting adequate, high-quality sleep consistently. This means going to bed at a reasonable hour, creating a sleep-conducive environment that's dark, quiet, and comfortable, and allowing enough time for complete sleep cycles to occur. Consistency matters. Going to bed and waking at similar times each day, even on weekends, supports your circadian system and enhances sleep quality. Avoiding alcohol near bedtime is important for recovery. While alcohol might help you fall asleep faster, it disrupts sleep architecture, reducing REM sleep and deep sleep, the very stages prioritized during recovery. A glass of wine with dinner several hours before bed is unlikely to be problematic, but drinking in the hours before sleep directly interferes with recovery. Creating a wind-down routine helps transition to sleep. This might involve dimming lights, avoiding screens, reading, taking a warm bath, or practicing relaxation techniques. The goal is to signal to your body and brain that sleep is approaching, allowing the biological changes that promote sleep to begin. For people dealing with chronic sleep restriction rather than acute deprivation, recovery requires not just a few good nights, but sustained behavior change. If you've been sleeping 5 to 6 hours nightly for months or years, recovering might take weeks of consistently getting 7 to 8 hours. During this extended recovery, you might initially feel more tired, not less, as your brain attempts to catch up on accumulated sleep debt. This can be discouraging, but represents your body's attempt to restore itself. Persist through this period, and sleep quality and daytime function will eventually improve. Medical conditions that interfere with sleep, such as sleep apnea, insomnia, or restless leg syndrome, must be addressed for recovery to occur. No amount of time in bed helps if the time is spent struggling to breathe, fighting to fall asleep, or being awakened by uncomfortable sensations. Professional evaluation and treatment of sleep disorders is essential for anyone experiencing persistent sleep difficulties. The message of this section is both hopeful and demanding. Hope comes from your body's remarkable capacity to recover from acute sleep deprivation. The effects, while substantial, are typically reversible. Demand comes from the recognition that recovery requires commitment. You must actually get adequate sleep consistently, not just once, but night after night. Recovery isn't passive. It requires actively prioritizing sleep, creating conditions for good sleep, and protecting sleep time against the many demands competing for those hours. As we move into our final section discussing practical strategies, the recovery timeline we've explored here provides crucial context. Understanding how long recovery takes helps set realistic expectations and underscores why preventing sleep deprivation in the first place is so much more effective than trying to recover from it repeatedly. Prevention is easier than cure when it comes to sleep. Practical wisdom: recognition, protection, and integration. After journeying through the multifaceted impacts of one bad night, from cognitive fog to emotional turbulence, from metabolic chaos to immune disruption, from cardiovascular stress to impaired performance, and understanding both the neural mechanisms driving these changes and the recovery process, we arrive at perhaps the most important question: what do you actually do with this knowledge? Understanding what sleep deprivation does to you is valuable, but translating that understanding into practical wisdom that shapes your decisions and behaviors is what actually matters. This final section brings together key insights and provides actionable strategies for recognizing sleep deprivation's effects, protecting yourself from its consequences, and integrating sleep health into your overall approach to well-being. Recognition represents the crucial first step. One of the most insidious aspects of sleep deprivation is how it impairs your ability to recognize that you're impaired. The disconnect between subjective perception and objective performance means you might feel reasonably functional while actually operating at significantly degraded capacity. This perception gap is dangerous. You make decisions as if you're operating normally when you're not. Learning to recognize the signs of sleep deprivation in yourself requires attention and honesty. Physical signs include persistent yawning, heavy eyelids, difficulty keeping your eyes open, the need to rub your eyes frequently, and slowed movement. Cognitive signs include reading the same paragraph repeatedly without comprehension, losing your train of thought mid-sentence, struggling to find common words, and increased errors in tasks you normally perform easily. Emotional signs include feeling more irritable than the situation warrants, experiencing disproportionate emotional reactions to minor events, difficulty controlling your emotional responses, and feeling more anxious or depressed than usual. Behavioral signs include increased clumsiness, difficulty making decisions, struggling with tasks requiring sustained attention, and a strong desire to consume caffeine or sugar to maintain alertness. When you notice these signs, the appropriate response is acknowledging impairment and adjusting accordingly. This might mean postponing important decisions, asking for help with demanding tasks, avoiding activities that could be dangerous if your attention lapses, or simply being more careful and deliberate in your actions. The key is recognizing that your current state isn't normal, even if you feel like you're managing adequately. For critical tasks like driving, the stakes are particularly high. Drowsy driving kills thousands of people annually, and the cognitive and attention impairments after poor sleep directly contribute to this risk. If you're drowsy while driving, symptoms include difficulty maintaining speed, drifting between lanes, missing exits or road signs, yawning repeatedly, having trouble keeping your eyes open, or not remembering the last few miles driven. Any of these warrant immediately finding a safe place to rest. The only safe response to drowsy driving is stopping and napping or having someone else drive. No amount of loud music, cold air, or caffeine fully compensates for severe sleepiness while driving. If you must drive after poor sleep and feel alert enough to do so safely initially, plan shorter trips, avoid monotonous highways where possible, and be prepared to stop if drowsiness emerges. The risk is real, and acknowledging it could save your life or someone else's. For professional contexts, recognizing impairment means communicating limitations when appropriate. In high-stakes professions where others' safety depends on your performance, such as healthcare, aviation, or emergency services, protocols should exist for reporting fatigue and being removed from safety-critical duties. Using these protocols isn't weakness. It's professionalism and responsibility. In less critical contexts, recognizing impairment might mean requesting deadline extensions, delegating tasks requiring peak cognitive function, or simply being honest with colleagues that you're not at your best. Most reasonable people understand that we all have off days, and transparency about your state often generates more respect than attempting to hide struggles. Protection strategies fall into two categories: preventing sleep deprivation when possible and mitigating its effects when it occurs. Prevention is always preferable. Prioritizing sleep means treating it as non-negotiable rather than optional. This requires setting boundaries around work, social commitments, and entertainment that encroach on sleep time. It means having a consistent sleep schedule and defending that schedule against demands to stay up late or wake early. Creating a sleep-conducive environment is essential for prevention. Your bedroom should be dark, cool, around 65 to 68 degrees Fahrenheit, and quiet. Investing in blackout curtains, a comfortable mattress and pillows, and perhaps white noise or earplugs, if environmental noise is unavoidable, can dramatically improve sleep quality. These aren't luxuries. They're basic sleep hygiene practices with demonstrated benefits. Screen time before bed deserves special attention. The blue light emitted by phones, tablets, and computers suppresses melatonin production and can delay sleep onset. Using blue light filters, wearing blue-blocking glasses in evening hours, or simply avoiding screens for an hour or two before bed all help protect sleep. The content matters too. Engaging, emotionally arousing, or stressful content makes falling asleep more difficult. If you must use devices near bedtime, choose calming, undemanding content. Alcohol and caffeine deserve careful management. Caffeine has a half-life of about 5 to 6 hours, meaning that half the caffeine from a cup of coffee consumed at 3 p.m. is still in your system at 9 p.m. For many people, caffeine after early afternoon interferes with sleep onset or depth. Finding your personal caffeine cutoff time and respecting it protects sleep. Alcohol, as mentioned earlier, disrupts sleep architecture even if it helps you fall asleep initially. If you drink, doing so several hours before bed minimizes impact on sleep quality. When sleep deprivation does occur despite your best prevention efforts, mitigation strategies help reduce the impact. Strategic napping provides the most effective acute intervention. A 20 to 30-minute nap can substantially improve alertness and performance for several hours. The ideal time for such naps is typically early to mid-afternoon, when circadian rhythms naturally promote a dip in alertness. Napping too late risks interfering with nighttime sleep. Longer naps, 60 to 90 minutes, allow complete sleep cycles and may provide more extensive benefits, but require more time and risk sleep inertia upon waking. For severe sleep deprivation, a longer nap might be warranted despite these trade-offs. Setting an alarm prevents oversleeping, and creating a comfortable nap environment, even just reclining in a quiet space with eyes closed, helps maximize the benefit. Caffeine used during sleep deprivation requires thoughtful timing. Consuming caffeine when you first notice drowsiness provides benefits when you most need them. However, caffeine consumed too late in the day interferes with recovery sleep that night. A practical approach is using caffeine strategically in morning and early afternoon, then avoiding it thereafter to allow recovery sleep to occur on schedule. The combination of napping and caffeine, sometimes called a coffee nap, shows interesting synergistic effects in research. Consuming caffeine immediately before a 20-minute nap might seem counterproductive, but caffeine takes about 20 to 30 minutes to reach peak blood levels. You nap before the caffeine kicks in, and you wake as it's taking effect, potentially getting benefits from both interventions. Some research suggests this combination improves alertness more than either alone. Light exposure helps manage drowsiness and supports recovery. Bright light exposure, especially morning light, enhances alertness and helps anchor your circadian rhythm. Even on days after poor sleep, getting outside or sitting near windows in morning hours helps your body understand it's time to be awake and begins the recovery process. As evening approaches, dimming lights and avoiding bright screens supports the evening rise in melatonin that promotes sleep onset. Physical activity during sleep deprivation requires balance. Gentle movement, like walking or stretching, can improve alertness without imposing significant additional stress on your already challenged system. Vigorous exercise might feel harder than usual and could increase injury risk given the coordination impairments we've discussed. If you exercise regularly, reducing intensity and duration after poor sleep might be prudent, prioritizing recovery over performance. Social support and honest communication help manage the interpersonal consequences of sleep deprivation. Letting family members know you didn't sleep well helps them understand if you're more irritable or less patient. Warning colleagues that you're running on limited sleep might make them more understanding of slower work pace or requests for help. You don't need to dwell on your sleep loss, but brief acknowledgement can prevent misunderstandings. For chronic sleep restriction, where you're consistently not getting adequate sleep week after week, more substantial changes are likely needed. This might require examining your schedule and commitments to find where time can be reclaimed. Many people find that honest assessment reveals activities consuming substantial time that don't truly enhance their lives. Reducing or eliminating these activities can free time for sleep without sacrificing things that genuinely matter. Work schedules present particular challenges. If work demands consistently prevent adequate sleep, conversations with supervisors about workload, deadlines, or schedule modifications might be necessary. Not all workplaces will be receptive, but some will, especially when you can articulate how sleep deprivation affects your performance and how adequate sleep would benefit both you and the organization. For parents of young children, sleep deprivation often feels unavoidable. While newborns do require frequent nighttime care, strategies exist to minimize the impact. Sleep when the baby sleeps, even if this means napping during the day. Share nighttime duties with a partner when possible. Accept help from family or friends who offer to watch the baby so you can nap. And know that this phase, though exhausting, is temporary. Sleep patterns typically improve as children age. Shift workers face unique challenges as their work schedules conflict with natural circadian rhythms. Strategic napping before shifts, using blackout curtains and sleep masks to create darkness for daytime sleep, and carefully timing caffeine and light exposure can all help. Some shift workers find that maintaining a consistent, if unconventional, sleep schedule, even on days off, helps their bodies adapt. Others prefer reverting to normal schedules on days off, accepting short-term disruption for better quality time with family and friends. Medical conditions interfering with sleep absolutely warrant professional attention. If you consistently struggle to fall asleep despite adequate time and sleep-conducive conditions, you might have insomnia requiring treatment. If you snore loudly, wake gasping, or experience daytime sleepiness despite seemingly adequate sleep time, sleep apnea should be evaluated. Restless leg syndrome, periodic limb movements, and numerous other conditions can disrupt sleep. These aren't problems you should try to solve alone. Sleep medicine specialists can diagnose and treat these conditions effectively. Technology offers tools for sleep tracking and improvement. Wearable devices and phone apps can track sleep duration, timing, and sometimes even sleep stages. While not perfectly accurate, they provide useful data about your sleep patterns over time. This data can reveal trends such as consistently inadequate sleep on certain nights or disruptions you weren't aware of. Some apps offer sleep coaching, relaxation exercises, or bedtime reminders to support better sleep habits. However, technology can also harm sleep. Obsessing over sleep metrics, a phenomenon sometimes called orthosomnia, can create anxiety about sleep that paradoxically makes sleep worse. Use technology as a tool, not a taskmaster. If tracking your sleep creates stress, stop tracking and rely on how you feel and function as measures of sleep adequacy. The broader integration of sleep into your health philosophy requires elevating sleep's priority. Western culture often treats sleep as wasted time, something to minimize to make room for more productivity, more entertainment, more of everything except rest. This perspective is fundamentally misguided. Sleep isn't the absence of productive activity. It is productive activity. During sleep, your body and brain perform essential maintenance, restoration, and optimization that enable everything else you do while awake. Reframing sleep as an essential pillar of health, equally important as nutrition and exercise, helps justify the time investment. You presumably don't feel guilty about eating or exercising. Sleep deserves the same status. When you get adequate sleep, you perform better at work, maintain better relationships, make sounder decisions, stay healthier, and experience greater well-being. The hours spent sleeping aren't stolen from life. They're the foundation supporting the quality of all your waking hours. Teaching younger generations about sleep's importance matters enormously. Children and adolescents need more sleep than adults, yet social pressures, academic demands, and screen time often prevent them from getting it. Parents who model good sleep habits, enforce reasonable bedtimes, and create sleep-conducive environments help their children establish healthy patterns that can last lifetimes. Schools that recognize sleep's importance and avoid unreasonably early start times, especially for adolescents whose circadian rhythms naturally delay during puberty, support student health and learning. At a societal level, normalizing and accommodating sleep needs would benefit everyone. Flexible work hours that allow people to work during their optimal circadian times, rather than forcing everyone into the same schedule, would improve both productivity and well-being. Understanding that someone who needs to leave a late meeting or decline a social engagement to protect sleep time isn't antisocial or uncommitted. Their being wise would reduce the social pressure to sacrifice sleep. Cultural shifts take time, but individual choices contribute to larger changes. The knowledge you've gained through this exploration of sleep deprivation's effects provides power. You now understand what's actually happening in your body and brain after poor sleep. You can recognize the signs of impairment in yourself and others. You understand why protection and recovery matter and have strategies for both. Most importantly, you have biological justification for prioritizing sleep, not as selfishness or laziness, but as an essential health behavior. One bad night affects everything. Your cognition slows, your emotions destabilize, your metabolism lurches, your immune system falters, your heart and blood vessels strain, your physical performance degrades, your brain operates under duress, but your body also knows how to recover. Given adequate sleep, consistently provided, your systems return to balance. The impairments resolve, the strain eases, the clarity returns. The choice is yours. You can treat sleep as optional, accepting the consequences we've explored, or you can honor your body's need for rest. You can push through exhaustion, operating at reduced capacity, or you can acknowledge that sometimes the most productive thing you can do is sleep. You can view sleep as wasted time, or you can recognize it as the foundation of everything you want to accomplish while awake. This isn't about perfection. You will have bad nights. Life happens. Stress occurs. Circumstances sometimes prevent ideal sleep. But those occasional poor nights needn't become the pattern. And when they do occur, understanding what's happening and how to recover empowers you to minimize the damage and bounce back effectively. Sleep is not your enemy, stealing time from important activities. Sleep is your ally, enabling you to perform those activities well. Treat it accordingly, and your body will thank you with clearer thinking, steadier emotions, better health, and greater capacity to engage with everything life offers. If you've made it this far, you now carry a depth of understanding about sleep's immediate importance that extends far beyond what most people know. You've seen how quickly and how dramatically your body responds to sleep deprivation, how interconnected all your systems are, and how fundamental sleep is to essentially every aspect of human functioning. Perhaps, after tonight, when you're tempted to stay up late for one more episode, one more hour of work, one more social media scroll, you'll pause and consider whether the trade-off is truly worth it. Perhaps you'll recognize those signs of impairment we discussed and make the wise choice to protect yourself and others by acknowledging limitations. Perhaps you'll defend your sleep time a bit more fiercely, knowing that those hours in bed aren't wasted, but are among the most valuable health investments you can make. Your body is remarkable. It can withstand considerable stress, compensate for substantial deficits, and recover from notable insults. But it does have limits, and it does require care. Sleep represents one of the most fundamental forms of self-care. Biological maintenance that cannot be outsourced or indefinitely postponed. If you found value in tonight's exploration, please consider subscribing to the Optimal Life Project and liking this video. Your support allows us to continue creating content that explores the science of human health and performance in depth, translating research into practical wisdom you can actually use. And most importantly, tonight or tomorrow night, or whenever you next have the opportunity, sleep. Sleep well, sleep enough, and wake ready to engage with life from a foundation of restoration rather than depletion. Your body knows what it needs. The question is whether you'll honor that knowledge. Thank you for listening. May your next sleep be deep, restorative, and sufficient. Good night.